Good morning, everyone. Respected dignitaries on and off the dais, distinguished scientists, academicians, researchers, faculty members, delegates, scholars, students, ladies and gentlemen. It is my great pleasure to welcome you all to Biospectrum 2026, the international conference on recent advances in biological sciences, bridging research to breakthroughs, organized by the Department of Bioscience, Macfast, in association with the KSCSTE JNTBGRI, the National Institute of Occupational Health, South Africa, and the Kerala Botanical Society, with the support of Kerala State Higher Education Council and DSIR. Over the next 2 days, this conference will serve as a platform for scientific exchange, innovation, and collaboration among researchers and academicians from diverse fields of biological science. On behalf of the organizing committee, I extend a warm welcome to all of you and wish you a productive and enriching conference experience. Before we begin, I would like to highlight the eco-friendly seed pens provided to all the participants as the Macfast Green Initiative 26. May I request everyone to kindly rise for a moment of silent prayer. Thank you. Please be seated. It is my privilege to invite our respected principal, Professor Dr. Varghese Kecharian, a distinguished academic leader whose guidance continues to strengthen the culture of excellence at Macfast, to deliver the welcome address.
Most respected Manager and Administrator, Reverend Dr. Thomas Kutte Pathinatil, the President of this function, Honorable Vice Chancellor of COFOS, Dr. A. Bijukumar, and the keynote speaker, Dr. Nandakumar Kalarikal, distinguished dignitaries on and off the dais, most respected national and international speakers, dear faculty members, research scholars, and students. A very good morning and a warm welcome to one and you. Today we are gathered here to make a fresh start to Biospectrum 2026, an international conference on recent advances in biological sciences. As principal of Macfast, it gives me immense pride and happiness to welcome you all to Biospectrum. Biospectrum is more than just a conference. It's a reflection of the dedication and collective efforts of our faculty, students, and the Department of Biosciences. Over the years, it has grown into cherished Macfast tradition, celebrating scientific excellence, innovation, and meaningful collaboration. Today, science is no longer confined to individual disciplines. From unlocking the therapeutic potential of tropical plants to engineering solutions at the nanoscale, from decoding the genetic blueprints of life to harnessing artificial intelligence for drug discovery, the science being discussed in of a new chapter as an autonomous institution. The introduction of new academic programs and our growing research initiatives reflect our commitment to excellence in higher education. Through international conferences, research collaborations, innovation-driven programs, and academic excellence, Macfast continues to emerge as a vibrant center of learning and a distinguished face of higher education in Kerala. My primary duty and responsibility is to officially welcome our dear guests. First, I would like to invite Reverend Dr. Thomas Kutte Pathinatil, our Manager and Administrator, whose constant support has been the backbone of every initiative at Macfast. May I welcome Father Thomas Kutte Pathinatil to preside over this function. Professor Dr. Bijukumar, the Honorable Vice Chancellor of Kerala University of Fisheries and Ocean Studies, a distinguished academician in the field of aquatic biology and fisheries sciences. Renowned for his pioneering contributions to biodiversity conservation, freshwater ecology, and sustainable fisheries management, Professor Bijumar has significantly advanced research and higher education in aquatic sciences. His visionary leadership and commitment to scientific excellence have strengthened Kufos as a center of innovation and knowledge. We are honored to have Professor Dr. Biju Kumar at Biospectrum Conference 2026 and look forward to his valuable insight and inspiring presence. May I welcome Dr. A. Biju Kumar to this august gathering. It is my pleasure to welcome Dr. Nandakumar Kalarikal, the Director of the International Center for Ultra First Studies and the former Director of the School of Physics and Applied Physics of MG University and the Director of Nanoscience and Nanotechnology of Mahatma Gandhi University. Professor Dr. Kalarikal, a good friend of mine and Macfast, has played a pivotal role in advancing interdisciplinary research and fostering scientific excellence. His dedication to innovation and academic leadership continues to inspire researchers and students across the globe. As we all know, is visiting to welcome him to the Biospectrum Conference and look forward to his valuable insights and contributions. I am delighted to welcome Dr. Ramesh Kumar Kebi and Dr. Shijulal, who is the backbone of this conference, and they will always be with us in Macfast for all the activities of this conference, and especially in the coming conferences in recent years. Dr. Ramesh Kumar is a member of the organizing committee, and also Mr. Shijilal, alumni of this Macfast College. May I heartily welcome Dr. Ramesh Kumar and Dr. Shijilal. I'm happy to welcome our international speakers, Dr. Rajiv Nair from Norway, Dr. Intan Safina from University of Malaysia, Aken Emmanuel Noki from the University of South Africa, Dr. Jitsi Joseph from Technomation University, South Africa. And we are deeply honored by the presence of these eminent scientists to this conference. A hearty welcome to our national speakers, Dr. Binod, Dr. Gopalakrishna Srijit from CSIR NIST, and Dr. V.B. Samir Kumar from Central University of Kerala, and Dr. Rajesh Raju from Enapaya University, and Dr. Brilliant Rajan from St. John's College. Your contributions to Indian science make your participation here truly meaningful. I also take this opportunity to extend my heartfelt appreciation, warm welcome to our conveners, Dr. Jenny Jacob and Dr. Tina Merlin, along with Dr. Ramesh Kumar Kebi, and to the entire team working under the guidance, including our coordinators, organizing committee members, research scholars, and the dedicated teamwork and tireless efforts over the past several months have been instrumental in making Biospectrum 2026 a grand reality. To our sponsors, Federal Bank, Cygene Biotechnologies, and Spectratrix, we are sincerely grateful for your generous support. And to every delegate, researchers, faculty members, students who has come here today, whether from across the world or from just down the road, you are the heart of this conference. I encourage you to engage fully and interact freely, make connections genuinely, and carry something meaningfully back with you when you leave. Once again, I welcome you all most warmly to Biospectrum 2026. Let us make these 2 days count for science and for the world that science is trying to serve. Once again, hearty welcome to each one of you. Thank you very much.
Thank you, sir, for your warm words of welcome and for setting an inspiring tone for this international conference. To provide us with an overview of Biospectrum 2026 and the vision behind this conference, I invite Dr. Ramesh Kumar KB, Senior Principal Scientist, KCSTE, JNTBGRI, to address the gathering.
Most Reverend Father Dr. Thomas Kutti Padinettil, Professor Dr. Bijukumar, Professor Dr. Varghese K. Cherian, Dr. Nandakumar Kalarikal, Dr. Shijulal, and other dignitaries on the dais and off the dais. A very good morning and once again welcome to this international conference on recent advances in biological sciences. So, as mentioned by the principal, the Biospectrum series of this international conference, Biospectrum 2026. The series has started since from 2007, and it is one of the premier academic events happening in the field of life sciences in Kerala. So, as mentioned, the conference explores a vast area from the conventional techniques to the most modern AI-driven sectors. So it deals with various technologies and methodologies that addresses global challenges such as climate change, biodiversity loss, food security, emerging diseases, microbial, biotechnology, and sustainable resource management, overall for the well-being of human life. So as you are aware, the life science sector is advancing on a much fast pace, and this conference brings together excellent speakers from all over the globe to disseminate the recent trends in the field of life sciences. Overall, we have more than 10, around 15 speakers, both from international and national level, and online as well as offline speakers are here. So the conference aims to enhance the professional development of young researchers and students particularly through networking and exposure to global research perspectives. So we have nearly 250 registered participants in this event, and around 90 research papers are being presented in this, are going to be presented in this international conference. So my humble request to the student and research community is to engage in scholarly interactions that will enlighten your future research endeavors. So each edition of this conference is being organized in association with various national as well as international agencies and institutions like RGCB. Previously it was like RGCB, DST, NABARD, ICMR, and so on. So many such prestigious institutions were part of this conference, and it's a proud privilege as a member of KCST JNTBGRI to be part of this international conference. So this year this program is being organized in association with KCST JNTBGRI, National Institute of Occupational Health South Africa, and Kerala Botanical Society. So in fact KCST JNTBGRI is having a long legacy of association, close association with Macfast. Macfast in fact has initiated some innovative courses related to various aspects of plant science like phytochemistry. The way back in around 15 years back, I think, my research guide and former head of the phytochemistry division, Dr. V. George, he was instrumental in designing the curricula of this plant sciences course. So I use this opportunity to fondly remember the contributions of Dr. V. George as well. So just a few words about KCST JNTBGRI. It is one of the largest conservatory botanic gardens in Asia. So we have nearly 4,500 species, 45,000 accessions of these species are conserved at JNTBGRI, and various aspects of plant science research like plant conservation, plant systematics, plant biotechnology, and then plant chemistry, of course, are being dealt in detail at JNTBGRI. So we have all the basic facilities for these advanced research activities of various aspects related to plant science research, and we are having an MOU, Memorandum of Understanding, in various research as well as academic collaborations with Macfast. And under this program, students are doing internship programs at JNT BGRI. Various collaborative programs are happening, and this conference is an example. So I also use this opportunity to mention a few words about Kerala Academy of Sciences. So KAS is also having a close association with Macfast. In 2024, I think we have organized an international conference in phytochemistry in collaboration with the KAS. So as the former General Secretary of Kerala Academy of Sciences, it's my proud privilege to stand here in this conference as well. So today we are having Professor Dr. A. Bijukumar, Honorable Vice Chancellor of KUFOS, Kerala University of Fisheries and Ocean Studies, Kochi, with us. So aquatic environment, particularly that of marine ecosystem, is right now well aware. Almost all of you are well aware of the details of marine environment particularly. And the credit goes to the efforts mainly by Professor Dr. Bijukumar sir. So he is such an ardent researcher in this field of marine environment systems, particularly with so much of research publications, journal articles, public programs, seminars and so on. Such a very dynamic personality, and we are very lucky to have him as to inaugurate this international conference. So thank you very much, sir, for gladly accepting our invitation for this function. And also, as I mentioned, this field of life science is a rapidly evolving sector. And just now, Professor Dr. Nandakumar Kalarikal was mentioning about the recent developments, like from nanoseconds to femtoseconds, it has been evolved. And we are having all these facilities in the nearby MG University, Mahatma Gandhi University campus, the International Center for Ultra-Fast Studies. So these are the recent happenings that can be assimilated to various R&D activities in life sciences as well. So we are also happy to have you, sir, Professor Nandakumar Kalarikal. And my friend Dr. Shijilal is there. So I welcome all of you once again, and particularly the participants. Wish you a very pleasant stay for 2 days, and moreover a very insightful and enlightening scientific discussions happening for these 2 days. So thank you all.
Thank you, sir, for presenting the objectives and significance of the conference so eloquently. May I now respectfully invite Father Dr. Thomasooty Padanettil, Manager and Administrator, Macfast, to deliver the presidential address.
Respected dignitaries of the dais, Professor A. Bijukumar, Dr. Nandakumar, Dr. Shijilal Nelson, Dr. Ramesh Kumar, our principal, Dr. Varghese Kecharian, Dr. Jenny Jacob, Dr. Basil George, Dr. Tina Merlin, distinguished academicians, scientists, researchers, faculties, and my dear students, good morning. Good morning. It is my great honor and privilege to address you all to this international conference on recent advances in biological sciences, Biospectrum 2026. On behalf of the Department of Biosciences in Macfast and the organizing committee, I extend a warm greeting to every participant who has joined us from different national and international research institutions, departments, regions, and countries. My best wishes to all our speakers, invited experts, sponsoring organizations, collaborating institutions, and my special appreciation to all the members of the organizing team whose hard work and dedication made this conference possible. This conference provides a unique platform for researchers, academicians, scientists, scholars, and students to exchange ideas, share findings, and foster collaborations. The various lectures, panel discussions, and poster presentations, we hope, will stimulate meaningful dialogue and inspire future research endeavors. As we know, science has shown its capacity to respond rapidly and efficiently, effectively to global challenges. And especially, biological sciences today stand at the forefront of innovation and discovery. This century has witnessed remarkable breakthroughs in genetics, biotechnology, microbiology, environmental sciences, bioinformatics, neuroscience, and medical research. These advances are transforming our understanding of life and providing powerful tools to address some of the world's most pressing challenges, including disease prevention, food security, environmental sustainability, and climate change. Today, to our young researchers and students who are here, I would like to offer a special word of encouragement. You represent the future of biosciences. Your curiosity, creativity, and dedication will drive the next generation to discoveries and experiments. I encourage you to actively participate in the discussions, engage with experts, and use this opportunity to broaden your perspectives and build lasting professional networks. I also thank each participant for contributing your expertise and enthusiasm to this important event. May our discussions lead to new insights, productive partnerships, and solutions that contribute to a healthier, more sustainable, and prosperous world. I wish this conference, Biospectrum 2026, every success and all participants an enriching experience here in Macfast. Thank you.
Thank you, Father, for your insightful and encouraging message. The ceremonial lighting of the lamp symbolizes the triumph of knowledge over ignorance and marks the formal commencement of this academic event. May I request our esteemed dignitaries to kindly proceed for lighting of the lamp. Thank you, dignitaries. It is now my privilege to invite Professor Dr. A. Biju Kumar, a renowned aquatic biotechnologist, academician, and researcher currently serving as the Vice Chancellor of Kerala University of Fisheries and Ocean Studies, Kochi. With an exemplary academic career spanning over 3 decades, he had made significant contributions to the fields of aquatic biodiversity, fish taxonomy, conservation biology, and inland fisheries. Prior to assuming the office of Vice Chancellor, he served as a professor and head of the Department of Aquatic Biology and Fisheries at the University of Kerala. As an accomplished scientist, Dr. Kumar has authored numerous research publications, books, and book chapters, and has successfully guided several doctoral scholars. His research has greatly contributed to the documentation and conservation of freshwater fish diversity in the Western Ghats, a global biodiversity hotspot. Recognized nationally and internationally for his scientific contributions, Dr. Kumar continues to promote excellence in fisheries education, biodiversity research, sustainable resource management, and innovation at aquatic science through his leadership at KUFOS. I warmly welcome Sir to deliver the inaugural address and officially inaugurate Biospectrum 2026.
Thank you. Thank you and good morning. Good morning to all of you. Respected President of the function, Father Dr. Thomas Kutti Padanettil, President of this college, this institution, Professor Varghese Cheriyan, and my friend Dr. Ramesh Kumar. Invite the keynote speaker today, Dr. Nandakumar, and the other members in the dais, dear friends, participants of this Biospectrum 2026. First of all, let me thank the college for inviting me to inaugurate this very important international conference. And I am also very much delighted to be here, primarily because of 2 reasons. One, there are some institutions which actually walk before time, and Macfast is one of the such institutions which designed some of the very unique programs for both graduation and post-graduation, and that is why Macfast stands different. And that is the reason why when Dr. Ramesh invited me for this conference, I immediately agreed, because I did not visit the institution, but I heard as an academician in this field in biological sciences for the last 3 decades, I heard this about this institution quite long through my friends, including the friends from TBGRA, Dr. Achyut Shankar, and all these people who actually got closely associated with this institution. And second reason was the title of this international conference, Recent Advances in Biological Sciences, Bridging Research to Breakthroughs. And it was absolutely relevant in this era, primarily when we speak about biological sciences. I would say rather life sciences. Because, if you speak about science in general, I am speaking to you for the next 10 minutes primarily as a biologist and as a person, as a science communicator, and just tell you what are the relevance of the life sciences or biological sciences at the particular moment, or in the particular moment in the history of sciences as per se. And we always say that the 20th century belonged to physics and computing, and I would say 21st century primarily belongs to life sciences. And there are specific reasons why I say that, because science is no longer the kind of volume of data you generate, but it's basically, and also the number of papers you publish, but it's basically the output of the science, which ultimately cater to the needs of the society. And that is why life science is more important in the recent decades. For example, you look at some of the very important demands of our time, for example, healthcare. And everybody now speak about, especially in the last 1 week, primarily we discuss only about health and what are the kind of diseases emerging, especially in a state which is always proud that we are actually on par with the developed nations in terms of our health standards At the same time, in terms of emerging diseases, Kerala is a hotspot. In terms of lifestyle diseases, Kerala is a hotspot. Despite the fact that we have wonderful health facilities here and wonderful health care system here, but the problem is that we are still prone to many diseases, especially the emerging diseases in Kerala particular. And second is that the main second question definitely is the food security. Now, if you look at the world population and the kind of degradation of the agricultural ecosystems, and we always think about food security, and food security always is connected with the life sciences, and food security is always connected with the way we how going to produce the agricultural produce using technology. For example, if you look at the entire history of human civilization, and I'm sure that as biologists we are all aware that we have a very short memory, and that's a very especially historical memory is very, very short for us. We speak, for example, if you look in terms of politics, we speak only about last 5 years, what the previous government did, and not in terms of hundreds of thousands of years. But as a biologist, I always speak about millions of years, because that's actually our evolutionary history. And if you look at the human evolution and the human history, it's almost 300,000 years back. The first human being started walking on the surface of the earth as Homo sapiens almost 300,000 years back. And all our religion, all our the cultural civilization all came in the last 5,000 years back. And then you look at the entire history of human civilization and the way the human beings occupied different continents across the earth, and then the contemporary human society, we would see that there are only few challenges human species always faced. And one of the challenge is definitely food security. And if you ever speak about the reason why the initial human beings started their journey out of Africa, and that is also linked with the climate change. It's always food security that demanded human species to move out of the continents. And that now science tells us the story. And when the human beings occupied different parts of the world, and then they found out that we need not actually remain as nomadic people, we can settle somewhere because we can do farming. And that is only 20,000 years back, we realized that we can settle somewhere along the river basins and we can do agricultural practices. Again, the scientific discoveries played a very, very important role. They realized that there are some fertile soils along the world where you can do the farming and settle down. And then we got a lot of free time. The human beings, as the Homo sapiens story by the Harari, his book tells us, human beings got a lot of time. When we get a lot of time, what will you do? We do all the other sort of things. We love music. We start different kind of practice different kind of dances, etc. That means that is the point in which the cultural evolution also happened along with the human evolution. And then you understand that the major challenge at that period of time was actually famine, when the people started agricultural farming, and there was issues with the soil degradation, there was issues with the food scarcity, etc. And then for example, you look at the Indian famine. The largest number of people were killed in India because of famine, and now we realize that it is not actually the famine per se, but it is all political decisions. When the government at that period of British government who ruled India at that period of time, especially at the beginning of the 20th century, they decided that there could be a third world war, and whatever may be the stock available in India, agricultural produce, they exported to the Europe, and then the famine became very worse in India, and large number of people killed. And the human beings, the first challenge was food security, and we overcome the challenges primarily because we did science, we practiced science in farming. And that happened through Green Revolution. We used all the kind of modern hybridization technology. We used the wild variety, crossed it with a better yielding variety, and we produced the hybrid varieties. And that was the reason why a country like India, which was actually waiting for the ships to come, just to satisfy our culinary requirements, we are now exporting food rice, and that is primarily because we used science in agricultural practices. So science play a very, very important role in achieving food security, and that will continue in future also. That was the first challenge. In all the 300,000 years of human history, that was the first challenge. How do we find out the food material? The answer was very simple. We used science to solve the problem of food security and saving the people from death. And the second challenge, probably you can all reconnect, and second challenge was the diseases. And the first, the zoonotic disease, the first disease which killed a large number of people in the world was plague. And hundreds and thousands of people were killed across the world, especially in Europe. And we could not actually save many of the human beings at that period of time because there was no vaccine. There was no vaccine. And now, after COVID, in 1 week time, we had a vaccine. And we had a vaccine primarily because the scientists were working on the same kind of pathogen, especially the viral pathogen. And the first, the technology actually came from the Oxford University, and because the scientists, and the technology was given free. And that is the reason why, and the technology was not simple, we need not actually have to wait for long period of time, because we have a very interesting mRNA molecule, and with mRNA molecule, we can actually produce a vaccine. So we have bioinformatics, we have the biotechnology solution offered in the form of a vaccine, and that is the reason why we could save a large number of people from death. And again, the second challenge, the diseases, we could manage that primarily because we used science for the betterment of the society. And also one more thing happened, the technology for production of vaccine from mRNA was given free. It was not patented, it was given free, and that is the reason why India never invented the technology, and maximum amount of vaccine production happened in India, especially with the Serum Institute of in Delhi, and that is the reason why India exported huge quantities of COVID vaccine and supplied it to different parts of the world. And again, you can see there are two major challenges which is faced by the human beings in the entire history. One was food security, which was overcome through application of science, and second was actually the diseases, especially the microbial diseases, and we could manage that primarily because we used science in vaccine production. So you can see that this is the history of the world. This is the history of the world. We have to use science, especially if you focus on the future of the mankind, future of the planet. And now we have a large number of other challenges. One definitely is the declining agricultural production. We need to use technology. And second is the healthcare, and also making the planet suitable for life, especially if you think about the planetary health, etc. There also we need science in a larger manner. And the third is the healthcare and environmental sustainability, and also the planetary survival. In all these fronts, we need to use science. And in centuries, there was actually a habit, and that habit is still there in many of our university institutions, departments and colleges, where the people from one department never talk to the other department, and one lab never work with the other lab. And that culture is disappearing, my dear friends. You also should understand. And in future, after 20 years' time, if I have an opportunity to come here or in any other college, we will not have any biology department and botany and zoology department. We will only have life sciences departments, primarily because the boundaries between the subjects are disappearing like anything. And I have few examples, and you go through some of the Nobel Prize winning discoveries in the recent past and connect it with the life sciences, and then you will understand that how important is the interdisciplinary science or transdisciplinary science. For example, and one classical example I always tell is about the Indian-born the scientist called Venkataraman Ramakrishnan. And basically he was a physics. He has his post-graduation and PhD in physics from India. And then his topic was basically crystallography study, if I remember correctly. And then he got interested in, and then he tried to apply his the knowledge in physics and the techniques he has, especially on crystallography, in understanding biological phenomena. And then he went abroad and then started learning biology. And then he worked specifically on ribosomes, understanding the structure of ribosomes. And then he studied the chemistry of ribosomes using all the kind of the learnings in physics that he had. And ultimately he has won the Nobel Prize in chemistry, and that is the way in which science is progressing. That means a man who has his basic graduation in physics migrating to biology because he wanted to apply the techniques in biology, and then he worked on specifically the chemistry of the ribosomes and won a Nobel Prize in chemistry. And that is a classical example how the modern science work. And as biologists, we should also understand that if you really wanted to understand the complex the phenomena in biology or even in biotechnology, definitely the science, especially the chemistry, play a very, very important role. And that is the reason why biological sciences or life sciences is becoming more and more interdisciplinary. And that is very, very important. And so that is happening across the world, especially if you apply for a institutional funding in the recent past. It's very, very important to understand that if you apply for example, you have a biotechnology department, bioinformatics department here, and if you apply only with a very basic the science in biotechnology and apply for a larger national funding, it won't work in the recent past. You need to actually work with other departments and make your science more interdisciplinary and work on an interinstitutional project and then apply, and then you have a more chance to get the project. And the younger students who are here, who are pursuing your career in graduation or post-graduation, you should also understand that, creating a question which demand an answer with an interdisciplinary research that will have a more and more demand in future. And that also you should remember understanding or working with the basic sciences in biology would not give you any opportunity for funding. So it's very, very important that you think interdisciplinary, think out of the box and come out with some of the novel ideas. And the second way in which I look at biological sciences is it's, if you really wanted to see God, immerse deeply in biological sciences. For example, again I will go to another Nobel Prize discovery, another one of my favorite, especially when I speak about science, and it is Svante Pääbo. And Svante Pääbo, he started his work, understanding the DNA structure of the human beings. And he has won Nobel Prize in physiology in the year 2022. And his research is very interesting, and I think you should read many, especially those who are studying biology, should read more books, primarily in the books which tells you the story of the human beings, and especially you start reading the Harari's book on Homo sapiens, and some other books which is which describe human evolution and history. And then you will understand that, you look at the Svante Pääbo's work, he primarily worked on ancient DNA. Ancient DNA means, DNA found in the fossils. And he developed a technology to isolate the DNA molecule from the human skeletons, which is thousands of years old. He collected all the ancient human fossils, including the Homo sapiens and other the hominids present in different parts of the world in the form of fossils. He developed a technology to isolate DNA from the human skeletons. And even human skeleton, if you look at internal ear or the joints between the skeletons, you may have some remains of DNA molecules, and it may be smaller fragments. And he developed techniques to copy it in larger numbers using, and also decontaminate the DNA molecule, and perfected the technology of isolating ancient DNA. And then he aligned all the DNA which he collected from different Homo sapiens and related the fossils from across the world. And he could found that very interesting phenomena that when the Homo sapiens moved out of Africa, they had a life with Neanderthals, the earlier human species, and then they have a hybridization, and that is the reason why even now the Homo sapiens carry at least 1 to 2% of the genome of Neanderthals. And what is the importance of that? One, it tells us a story that the human beings, now the story is very clear, because now we have the ancient human skeletons or fossils from across the world, and we have DNA from all these human fossils, and now the story of the human beings migrating from Africa, reaching different parts of the world, including our own Andaman and Nicobar Island, is very clear. And how the human beings originated, how they migrated to different parts of the world. And now the theory also tells us, the study also clearly tells us that how the human beings got our DNA contaminated with the Neanderthal DNA, because we, the Homo sapiens, also have some kind of hybridization with the Neanderthals. And what is the significance of this 2% of genome? 2% of the genome is responsible for many of the immunity issues which human beings faces. Many of our immunological problems, many of our disease connectivity and disease, the resistance, everything is, connected with the 2% Neanderthal genome. And that means that's very, very important. How our DNA react with the molecules, specific antibiotics, everything is very clear. And that is the reason why now the science is becoming more and more interdisciplinary. And the sciences tells us a lot of story, primarily because we now believe that every human being in the world, in the planetary scale, remain connected. We are all relatives in some way or other, and that relation comes only through the molecule called DNA. Okay, so that means you don't have to fight with in the name of religion, caste and creed, because we are all connected by a single molecule, and that molecule is DNA. And the DNA can work a lot of wonders in that sense. And that is the reason why the science is also becoming very, very interesting. And then along with the science, and that is more important, for example, there are scientists like David Leakey. His books are also very, very interesting. And he connects the migration of the human beings along with science and along with the linguistics, along with the culture, the people working in culture, and then they draw the connectivity. How do the languages originated? How do the cultures originated along with human migration? And they connect it exactly with the positions where the human being settled. And that kind of connectivity tells us more stories, more precise scientific stories about our own journey and our own way when we look at future. And that is the reason why this is very, very important. The biological science is very, very important. And biological science in connection with other sciences, rather transdisciplinary sciences, tells us our own story about our own origin and our own cultural evolution, the origin of languages, etc. The entire story is rewritten with the science when they work in interdisciplinary platforms. And that is the reason why it's very, very important. And when you have an international conference on biological sciences, recent trends, my dear friends, this is very, very important. And science becoming interdisciplinary, biological sciences becoming interdisciplinary to solve many of the problems that the human beings face. And that means we need to actually reorient our science in such a way to solve some of the pressing problems of the society as well. And that is why the biological science is very, very important. And that is the reason why 21st century is called the century of the life sciences. And one more thing, now we can, for example, you look at synthetic biology. You can design everything now. For example, if you have a problem with your nose or ear, everything we can actually create within the laboratory with the information that we have. So synthetic biology, like the 3D printing, that technology that the MG University is now developing very fast, and you can design everything in the laboratory now. And the recently, the laboratory meat is also came into market. For example, you need not actually cultivate cattle. From the stem cells, you can create the beef within the laboratory. And now the Singapore has allowed the registration. Within the laboratory, you can create the beef, and then you can supply it to the market. And then you need not actually go for any kind of carbon-intensive farming of the cattle. So the technology is moving very, very fast, and science is moving very, very fast to solve some of the problems. And synthetic biology is one such kind of arena where you have a solution. And now also, as biological scientists, biologists, the role of multiomics. And for example, we have a large number of transcriptomics, proteomics, metabolomics, biomics. Everything is playing a very, very important role in answering some of the very important problems, and that is also very, very important. And then, as biologists, we cannot stay away, because artificial intelligence is right there in front of you. And in many of the biologists, including I basically work with biodiversity documentation, we also use a lot of AI technology primarily for documenting life from the depth of the oceans. AI play a very, very important role, and artificial intelligence can play a very, very important role. For example, I am basically from a marine science background, and now one of the major issue, for example, is locating illegal fishing across the globe. And now some of the very young students, who are working with artificial intelligence and machine learning, they came out with this technology to track the vessels, ships moving across the world. Sitting inside the laboratory using artificial intelligence, they can track every ship going for fishing in every part of the globe, and then they can track which is an illegal vessel, and all these things are possible primarily because of technology. And now the technology also demand that for example, America stopped import of shrimps from India because we are not using eco-friendly technologies for fishing. And some of the European unions now demand that we should know from where you catch the fish. This is called traceability. For example, then only we accept our produce. And how do you do that? We have absolutely no record. There also we need technology, artificial intelligence, machine learning. For example, if you are catching a fish from here, from the Kerala coast at a depth of 100 meters, you should know from where you catch. and that is called traceability. And that certificate is required. And there, only artificial intelligence can answer these kind of questions in creating a knowledge which is essential for international trade. That's very important. So that means, in every walk of life, biology is becoming very, very important. And I am sure that after COVID, we have a lot of answers to the questions, what we need to do. And we have an answer because we cannot do the things in isolation. For example, if you are actually trying to find out an answer for a disease problem, definitely you have to link it with the multiple reasons. For example, COVID. Even now there is a dispute. What is the source of origin of COVID? And we know that COVID is only a mild pathogen. And if you look at the UNEP's report, there are more than 250 zoonotic pathogens. And COVID is only a mild pathogen. And that means, there are more than 250 species which are more virulent than COVID. And reservoir host is the wild animals. That means, again, if you look at science, you cannot do the science in isolation. That means finding out the pathogen is not the answer to the problem. You need to know what is the reservoir host. You need to know why the disease spread. And the reason is very simple, because we have deforestation happening. And that is the reason why many of the flying foxes, or the so-called owl, which usually stay inside the forest, they are all coming close to the human habitation, primarily because there is environmental disturbance. And that means we need to understand the ecosystem, we need the health, we need to understand the human health, we need to understand the plant health, we need to understand the animal health. That is the reason why we have a new concept called One Health approach. And that means after COVID, we have the way in which we look at science is also changing. We need to have a holistic look at the life and also planet. That means planetary health is more and more important, and that is actually the lesson which we learned from the COVID time. And when we speak after the COVID, post-COVID era, we need to see that our research also need to be rescheduled to cater to the demands of the society as well. And that is the reason why biology is very, very important. When we look at another important way in which the education sector is transforming is actually the hope for entrepreneurship and innovation. And many of the universities are now starting startups and the entrepreneurship opportunities. And many of the youngsters who have an innovative idea, they should actually go into the innovation hub, and also try to see that how your idea can be made into a process or a product ultimately. And all these things are the changes in the post-COVID era. And when you look at the future, and you should understand that your future will not be defined not only only by what you know, but by your ability to learn continuously, and also ability to transform continuously, and also innovate continuously. So innovation and transformation of knowledge, multidisciplinary learning, all the things are very, very important when we look at future. So when I look at the Biospectrum and its title, this is very, very important, and it's very important to remember that breakthroughs always begin with a question. So as an youngsters, as graduate and postgraduate students here, and many of the younger researchers here, and keep questioning and try to find out new questions in the research arena as well. And then for the solutions, you go out of the box, think independently, think creatively, and then you will get an answer to the solution. And the future of biological sciences, I should say, as a biologist, it is very, very promising. The only thing is that you cannot work in silos. You need to work with other people, you need to work with other disciplines, and then you will have a larger answer to the question which you are always seeking. I am sure that this conference will give some of the very interesting, or trigger some of the very interesting thoughts, which ultimately you find a way in which you have to do the biological research in a disciplinary manner. I am sure that this conference will give you some of the very key questions to ponder upon, and also some of the very good collaborations where you can think for your future. And I am sure that once again I congratulate the Mar Athanasius College of Advanced Studies for organizing Biospectrum 2K26 and wish the conference great success. And with all your permission, I declare that this conference is inaugurated. Thank you very much.
Thank you, sir, for your inspiring inaugural message and for formally declaring Biospectrum 2026 open. Research is best celebrated when knowledge is shared. We now proceed to the release of the conference abstract book, a compliance of scholarly contributions from participants of Biospectrum 2026. I invite Dr. A. Bijukumar, Vice Chancellor Kufos, to release the conference abstract book and hand over to our principal, Dr. Varghese Kecharian. May I kindly request our esteemed dignitaries to kindly join the ceremony. Thank you, dignitaries. We congratulate the organizing committee and all the contributors for this valuable academic resource. Now, I invite Principal of Macfast, Dr. Varghese Kecharian, to present a token of appreciation to Dr. A. Bijukumar. Thank you, sir, for your time and efforts. Thank you, sir. We now move to the keynote session. It is my honor to invite Dr. Nandakumar Kalarikal, the Director of the International Center for Ultra Fast Studies at Mahatma Gandhi University, Kerala, India. He is also the Professor of Eminence in Nanoscience and Nanotechnology at Mahatma Gandhi University and visiting scientist at the International and Inter-University Center for Nanoscience and Nanotechnology at Mahatma Gandhi University. He is a Fulbright Fellow, Professor at Lorraine Institute of Jean Lamour, France, CNRS Professor with Institute of Light Matter, Claude Bernard University, Lyon, France. He is also Senior Professor, former Director, Chair of the School of Pure and Applied Physics, former Director, Joint Director, International and Inter-University Center for Nanoscience and Nanotechnology at MGU, former Dean, Faculty of Technology and Applied Science, and former Member Syndicate and Senate at Mahatma Gandhi University. He has published more than 450 research articles in peer-reviewed international journals with H-index of 70 and more than 17,000 citations. He has also edited more than 45 thematic scientific books published in Elsevier, Springer, CRPC Press, etc. He has supervised 30 PhD and recipient of many international and national research grants. I am truly honored to invite Dr. Nandakumar to deliver the keynote address.
Thank you very much for the detailed introduction. At the outset, I would like to thank the organizers of Biospectrum 2K26 and the International Conference on Recent Advances in Biological Sciences at Macfast, particularly to the principal, Professor Varghese Kecharian, and also the team Biospectrum 2K26. It's always a great pleasure for me to come back to Macfast because I've been closely associating with Macfast for the past many years. There are so many reasons, like I have good friends over here and good collaborators, and also your campus is very greenish, which makes me more exciting every time I come to this campus. So today, as we are progressing with this international conference on recent advances in biological sciences, I would also like to say very good morning to all the dignitaries on this dais and also off the dais, the delegates from different parts of the country, and also all the speakers across the globe as part of this very interesting program. So if I've been just going through, initially I was thinking that I'm supposed to deliver a keynote lecture, so I prepared a lecture, but then I realized that it is in a keynote address. So I was just going through the major themes of the conference, which includes a vast themes like phytochemistry, phytopharmaceutics, drug discovery and development, environmental sustainability and ecobiology, genomics, nanotechnology in biology and medicine, microbial interactions and antimicrobial strategies, biodiversity, taxonomy, AI-assisted drug discovery, so on and so forth. So my areas of expertise actually falls into 3 themes which have been mentioned here, like nanotechnology in biology and medicine. We are very active in this particular field, microbial interactions and antimicrobial strategies, and of course AI-assisted research, not exactly with the drug discovery, but we make use of artificial intelligence and machine learning in material science. I am basically a material physicist, mostly working in the field of nanoscience and nanotechnology, and this was mentioned by our honorable chief guest, Dr. Bichu Kumar, honorable vice chancellor of KUFUS, that it is, the subjects of science has become more cross-disciplinary and interdisciplinary. So my expertise is actually falls in this field. I learned lots of chemistry, I learned biology, medicine, so on and so forth, also computational physics and chemistry to a larger extent in all my research activities. So I'm sure that during this 2 days of conference, all the delegates will get a good exposure in all the themes mentioned in the brochure, and you will have the opportunity to interact with experts in this field. Coming to the basic concept of nanotechnology in biology and medicine, we basically focus in the field of tissue engineering scaffolds for wound healing, artificial skin, and then being a material physicist, I work on lots of quantum systems, particularly quantum dots as imaging, biological imaging systems. Focusing mostly on cancer cells, how to image the cancer cells, and also we work with the drug release, particularly for HIV, etc. Like we design the molecules with the polymeric systems, and the sustained release of this drug in the appropriate areas we make use of. And also, as it was mentioned earlier, that 3D printing has emerged as an important system. So along with electrospinning techniques, we also work with the 3D printed scaffolds for wound healing application. Also, we are now into artificial organs, particularly the external organs. So we make the custom-made, 3D printed artificial organs for that matter. And also I'm associating with a couple of people from Pushpagiri Research Center and Believers Hospital to make a 3D printed skull. When a patient is a human met with an accident and is having a head injury, we cannot replace the same kind of skull for the treatment. So we need to have custom-made skull parts for treating such patients. So I'm associating with a neurosurgeon for this kind of research activities. So there is a lots of there are lots of scope for people to make use of nanoscience and nanotechnology for biological applications. Few of the things I've already mentioned. So in fact, I don't know how many of you are familiar with the nanoscience and nanotechnology. Can you raise your hands, please? Yeah, at least few of you are familiar with the nanoscience and nanotechnology. It is the field, it is the science and technology of very small things. When I say that it is very small, it could be at the atomic level and the molecular level, so on and so forth. So we design these molecules or any such kind of system, material, which is having a dimension which is in the range of 1 to 100 nanometers is normally termed as a nanomaterial, and these type of nanomaterials we design in the laboratory for specific application. So we focus mostly on translational research these days to address energy security, water security, health security, and then food security. And also we are into environmental sustainability in terms of climate change. We are looking into different aspects of climate change, how the black carbon is evolving as an emerging player in global warming. So we have lots of association with the different institutions to look into that. And coming to the biological aspect, tissue engineering, we work with the polymer nanocomposites. All of you are familiar with the polymers. So every electronic gadget or anything you make use of today will have a polymer component. So we make use of certain polymer matrices, particularly which are biocompatible. So there are natural polymers, and there are also synthetic polymers. So we basically work with PCl, which is a synthetic polymer, but it is biodegradable, biocompatible. So our main focus is on such kind of polymers for biological, tissue engineering applications. So we incorporate different nanoparticles like zinc oxide, titanium dioxide, and also we have made use of other type of nanomaterials into this to make it as a tissue engineering scaffold. So these scaffolds are being used for wound healing applications. So some of our now developed polymer nanocomposites with the PCl or PVDF TRFE, that is another kind of electroactive polymers which will have a certain kind of electrical signal generation due to mechanical movement. So that will also facilitate the cell proliferation and wound healing processes. So we work this kind of aspects. So we develop the materials and then we apply this for wound healing applications. Particularly now we are focusing on diabetic wounds, which requires much more complex and custom-made scaffolds. So for that particular aspect, we make use of the 3D printed base onto which we put the electrospun fibers, and then we incorporate the antibiotics or the concerned medicines, and then we look into the sustained drug release for wound healing applications. This is one of the potential areas of my group. Many students have completed their PhD theses with us in this particular area. Now coming to other areas in which you can look into, that particularly cancer cells. So we work with different type of cancer cells, how to image these cancer cells. For that, and also we work with a different magnetic system. So this magnetic system could be used as a potential tool for magnetic hypothermia. So you target these magnetic nanoparticles onto the cancer cells and then apply magnetic field, and with the generation of the heat, these cancer cells could be killed. So it is a collective, a very specific targeting of the cancer cells without damaging the normal cells. So for that we need to image the cancer cells. So we make use of different type of quantum dots. Particularly we are into carbon-based quantum dots. So our motivation is that we need to make use of sustainable kind of nanoparticles. There are quite large number of nanoparticles. So we should look into the kind of toxicity which can generate. Therefore these are biocompatible. These materials what you select are biocompatible. And my group also works with dental doctors. We have developed novel dentures replacing the normal tooth. So these dentures are being incorporated with certain nanoparticles. We are mostly into transition metal oxides. So these transition metals are needed for the body. So even if it is leached out into the body, it is not going to harm the body. So that is the basic hypothesis we make. So for every research what we do, we make a hypothesis, and based on that hypothesis we design the systems. As it was already mentioned by Professor Bichu that AI and machine learning techniques could be effectively employed for various applications. So we are now making use of this kind of simulation studies, particularly to reduce the number of animals used in the experiments. So there is a protocol. I was a member in the ethics committee of the Pushpagiri Dental College. So we restrict the number of animals to be used in such kind of animal models. So to reduce this number, probably this kind of predictions will help. So we also make use of this kind of applications in biology to reduce the number of animals used in experiments. So you can have such kind of simulation studies to incorporate in your research activities. So every time new technology is emerging, and we really don't know how it is going to affect the entire society in the coming years to come. Right now we are talking about nanotechnology in various applications, such as I was mentioned that I'm working in the the area of energy security applications. So we developed electrode materials for fuel cells, batteries, and we designed these energy systems. And we are also into EMI shielders, where we make use of the polymer nanocomposite to restrict from other radiations which are affecting most of the very sensitive electronic gadgets. For example, many of you might be using more than one cell phone. If you open the cell phone, there is a metallic flange, that is an EMI shielder. So the weight of the your electronic gadget, for example, the batteries and this kind of metal components within that. So we are trying to replace these metal components with the polymer nanocomposites, which will become more light. So there are lots of issues when we talk about a sustainable research, whether these materials what we use, they are degradable in terms of environmental sustainability. Should we use more natural based kind of sources for this kind of research? So I'm seeing that most of you are undergraduate students and postgraduate students. As part of our education policy, research has become an important component. It is not that we develop certain in the lab, but how far we are going to be successful in translating this research to the society. That is very, very important. So government has different options, and you have better opportunities to become an entrepreneur. You can have your own companies to implement your ideas and come up with a product. So at the end of the day, we need to look at the Mother Earth in such a way that are we protecting the Mother Earth or are we exploiting the Mother Earth? So restrict the kind of chemicals what we use. So in my laboratory, we mainly focus on green chemistry. Though I am a material physicist, I make use of lots of chemistry, lots of biology, lots of medicine, lots of concepts are needed. So integrate all those things. So make use of the fact that how sustainable research we can adopt and how it can be translated. So translational research is of course a big challenge in the country, but we are much more better, particularly in terms of biological research or medical research. Like we can do the animal experiments without much trouble. So some of my collaborators in abroad, they are not able to do the animal models, so we do the animal models for them because we have more flexible kind of approach. We have the committee to look into that. So that is one of the reasons we are mostly into this kind of computational modeling to reduce the number of animals used in our experiments. So that during these 2 days, I'm sure that all the delegates will have a wonderful time. So make use of this time to interact with the speakers, make some connectivity with the speakers so the postgraduate students can pursue their research or PhD if you're planning for such kind of career in the future. So there are lots of opportunities. The world is open. Lots of fellowship opportunities for you people to explore that. So now every information is available at your fingertips. Just do a Google search. You identify the people, make a contact with them well in advance, and try to make use of a dissertation, master's dissertation or undergraduate dissertation with some of these leading people across the globe. And once they are convinced, they will write specific projects for the students. I normally do that. So funding is also very, very important, but at the same time opportunities are enormous, I should say, enormous. So you should make use of that. Planning your career is also important. It is not that you acquire a degree after 2 years or 3 years. Now the undergraduate programs are 4 years of duration as per the NEP guidelines. So whatever it is, you should excel in your field. It's not that all of you should go to research, but there are other areas in which you can explore if you are inclined to science. So being a person in the field of science, I always educate that you should retain or you should be in the field of science so that the society will be benefited in the coming years. So you are the youngsters are the backbone of the nation, and I'm sure that all of you will have some ideas to go ahead with your career. So plan a good career. You have nice faculty members here. They can be good mentors. Interact with them. And these opportunities, the conferences will give you a wider opportunity to see how the world is progressing in different areas. So you'll get some ideas, and that ideas could be a spark for your research. So I wholeheartedly welcome the delegates who have come from India or abroad to visit Mahatma Gandhi University if possible and see our facilities and all the facilities what we have established over a period of time are open to all the researchers across the globe. And also Kerala is a beautiful place, take some time to move around. Now it's a rainy season, it will be very wonderful to have a drive in the rain. And Kutanad is nearby, you have lots of backwaters, enjoy the weather, enjoy the conference, and make good networking and good friendships during this period. So I thank the organizers once again for this opportunity, and I wish all the delegates and the speakers to have a very nice time during these 2 days, and I wish all the very success to this particular conference, Biospectrum 2K26, and the International Conference on Recent Advances in Biological Sciences. And thank once again for this warm invitation to visit Macfast and to be part of this Biospectrum 2K26. Thank you very much.
Thank you, sir, for sharing your valuable insights and perspectives with us. Drawn from decades of excellence in research and academia have greatly enriched our understanding and set an inspiring tone for the scientific sessions ahead. I invite Father Dr. Thomasooty Padanittil to present our token of appreciation to Dr. Nandakumar Kalarikal. Thank you, sir, for your tireless efforts and contributions. We now move on to the presentation of certificates of appreciation in recognition of valuable contribution to Biospectrum 2026. I invite Dr. Nandakumar Kalarikal to certificate the awardees. For e-design and digital creative development, I invite Mr. Adityan Kushubukumar of BCA 2326 batch. For digital platform development of Biospectrum 2026, I invite Mr. Aaron Verghese, Mr. Anandu Pradeep. I invite Mr. Biju to certificate the awardees. I kindly invite Dr. Biju Kumar to certificate the awardees. Ms. Sonia Paul, Mr. Ivan Binoy. For event and broadcasting of Biospectrum 2026, I invite Mr. Anthony Tom. Thank you, sir. I now invite our guest of honor, Dr. Shijilal Nelson Sadi, Scientist E1 and Coordinator of Bioinformatics Facilities, RGCB, to address the gathering.
Dignitaries on the dais and dignitaries on the dais and off the dais, and my friends and researchers and students. Good morning to all of you. For me, it's always a homecoming. Actually, I'm an alumni of this institute. I graduated from the institute in 2006, and I noticed this Biospectrum started happening from 2007. And like Professor Biju previously mentioned, I was attracted to the institute because the way the institute or the center started doing things in a different manner. Because in 2000, I was the third batch of a bioinformatics student in MSc. And that time, there were not many colleges offering this course. And the uniqueness of this institute was the one thing which has attracted me to this institute. Then I noticed along the academics, there was some emphasis on research activities that was started, I think, from the beginning of this institute. And I'm happy to say, happy to see, happy to witness, still this is getting strengthened over the time. And I was one of the beneficiaries of such an effort. Actually, I graduated in 2006, then now currently I have a laboratory, bioinformatics laboratory at the Rajiv Gandhi Center for Biotechnology at Thiruvananthapuram. And mostly we are studying how does antibiotic resistance is happening in, like, priority bacterial pathogens. So that I will be explaining those things. So in a nutshell, what I wanted to say is I really thank the organizers and for me, like, giving me this opportunity to come over and address you and share my research experience. And I really wish all of you will make use of this couple of days interacting with all the international speakers and national speakers. And this will be very useful for you, and at least some of you will get some stepping stone into the real research problems after this meeting. I wish you all the best, and yeah, thank you.
Thank you, sir, for your encouraging and insightful remarks. I now invite Dr. Jenny Jacob, Head of the Department of Bioscience and Convener of Biospectrum 2026, to offer felicitation.
Respected dignitaries on the stage and off the stage, participants of the conference, students, faculty members, and my colleagues. So it gives me immense pleasure to celebrate the resounding success of Biospectrum 2K26, a vibrant gathering that fostered scientific excellence, innovation, and collaboration among budding researchers and academicians. The event is witnessing an enthusiastic participation of 231 delegates, which includes 160 offline participants and 63 online participants, representing a diverse academic community from across India and abroad. Only around 65 participants are from Macfast College itself. The participants comprise around faculty members, 14 faculty members, 72 research scholars, 106 postgraduate students, 29 undergraduate students, and this reflects the broad spectrum of talent and scholarly engagement that has characterized the event. The scientific sessions are enriched by 96 oral presentations, 24 poster presentations, while 92 are participations. A notable highlight of Biospectrum 2K26 was its wide institutional representation. Participants have joined in from Rochester Institute of Technology, Dubai, Madurai Kamraj University, Davangere University, Wales Institute of Science and Technology, Yenapoya University, Karunya Institute of Technology and Science, Jain University, Maharaja Krishna Kumar Sinji Bhavnagar University, Gujarat, SJJT University, Rajasthan, Sandeep University, Nashik, Vignan's Foundation from Guntur, then Malvanchal University, Indore, Central University of Himachal Pradesh, SRM Institute of Science and Technology, Kufos, University of Kerala, KUSET. Almost all the universities of Kerala are represented in this conference. And we also have participation from major institutes like NIT Calicut, Manipal Academy of Higher Education, Nurul Islam Center for Higher Education, Madras Christian College, Chennai, Dr. NGP Arts and Science College, Vilas Rao Deshmukh Government Medical College, Lattur. So in fact, the Biospectrum is a representative of India, I would like to say in that words. So a special note of gratitude is extended to our esteemed collaborators and supporters whose invaluable partnership significantly enhance the success and impact of Biospectrum 2K26. We sincerely acknowledge the collaboration and academic support of the National Institute for Occupational Health, South Africa, Jawaharlal Nehru Tropical Botanic Garden and Research Institute, JNTBGRI, Trivandrum, Kerala Botanical Society, and the Kerala State Higher Education Council. Their commitment to advancing scientific research, innovation and academic excellence provide tremendous strength to our initiative. We also express our heartfelt appreciation to our sponsors, particularly Federal Bank, Cygene Biotech, Spectratix, Associated Scientific and GeneFit Hub. So their contribution reflects the vital role of academia and industry collaboration in fostering scientific growth and empowering the next generation of researchers. So let us remember that every great scientific breakthrough begins with curiosity, is nurtured by perseverance, and flourishes through collaboration. The ideas shared, questions raised, and discoveries presented during this event are seeds for future innovations that may transform healthcare, biotechnology, environmental sustainability, and society at large. May this experience inspire every participant to continue exploring the unknown, challenging existing boundaries, and contributing meaningfully to the ever-expanding frontiers of science. Together, we have demonstrated that science is not merely the pursuit of knowledge, it is a collective endeavor to create a better future. Once again, I congratulate all participants and thank everyone who contributed to making Biospectrum 2K26 a truly memorable celebration of scientific excellence and learning and innovation. Thank you.
Thank you, ma'am. Now I invite Dr. Basil George, Assistant Professor, CMS College, Kottayam, to offer the felicitation.
Reverend Dr. Thomas Kutti Padinatil, Manager of Macfast. Dr. Ramesh Kumar KB, Dr. Nandakumar Karikal, other dignitaries on and off the dais, research scholars, students, a warm good morning to all. It's a great pleasure for me to be here, to be a part of this Biospectrum 2526. First of all, I would like to congratulate the Department of Bioscience, Macfast, and the organizing committee for successfully organizing this international conference. As the previous speakers have mentioned, in today's rapid changing world, the biological science play an important role in improving human health, protecting biodiversity, advancing biotechnology, and promoting sustainable development. This conference like this Biospectrum provide valuable opportunities for learning, sharing ideas, and building meaningful cooperations among researchers and academicians. I am confident that the scientific session, discussion, interactions over the next 2 days will be enriching and inspiring for all the young researchers over here. And I especially encourage all the students and research scholars to make most of these opportunities by learning from experts, exchanging your ideas. And as a representative from the Kerala Botanic Society, I am delighted to see such initiatives to promote the scientific inquiry and academic excellence. Once again, I extend my sincere best wishes to all the organizers, speakers, and participants of Biospectrum 2026, and it should be a grand success and contribute meaningfully to advancement of biological sciences and the betterment of the society. Thank you.
Thank you, sir. As we conclude the inaugural session, I invite Dr. Tina Merlin, Convener, Biospectrum 2026, to propose a vote of thanks.
Respected dignitaries on and off the dais, distinguished guests, esteemed speakers, faculty members, researchers, students, and dear participants attending this conference online and offline. As we conclude this inaugural session, it is my pleasant privilege to propose the vote of thanks. On behalf of Department of Biosciences, I extend a heartfelt gratitude to Professor Dr. Biju Kumar, the Honorable Vice Chancellor of KUFOS, for inaugurating this conference and inspiring us with his insightful address. Thank you, sir. We express our sincere appreciation to Dr. Nandumar, Mahatma Gandhi University, for delivering a thought-provoking keynote address that has provided valuable perspectives for all participants. Thank you, sir. Our special thanks goes to Dr. Shijula, and we are very proud of this alumni for delivering his address and helping us always and inspiring so that we can contribute to the scientific community in all ways. Thank you, sir. We are deeply grateful to Father Dr. Thomas Gutti Padanattil, manager, for his presidential address and his constant support and visionary leadership. Thank you, Father. We are also thankful to our principal, sir, Verghese Kichenrian, for his warm welcome and unwavering encouragement and being a strong pillar in organizing this conference. I would like to place on record our sincere appreciation to Dr. Ramesh, sir, who has been instrumental in the successful organizing of this conference. Thank you, sir. Our heartfelt thanks to Dr. Jenny Jacob and Dr. Basil George, who have always been support and helped us towards the success of this event. Today, we also had the privilege to honor the silent workers who had been instrumental. They are, again, we are very proud because all the silent workers are the students of Macfast. So as proud Macfastians, I would like you all to give them a big round of applause for supporting us. I extend my gratitude to all members of the organizing committee, my faculty team, advisory board, student volunteers, technical staff, media team, IT department, and every participant who has contributed to making this conference a reality. As we embark on the scientific sessions ahead, may this conference serve as a platform for meaningful discussions, fruitful collaborations, innovative discoveries. Thank you all for your presence, support, and enthusiasm. We wish everyone an enriching and productive Biospectrum 2K26. Thank you.
Thank you, ma'am. This concludes the inaugural session. We shall now break for tea and networking. The scientific session will resume at 11:30 a.m.
Hello.
Welcome back. We now move into the scientific session of Biospectrum 2026. The plenary session will be chaired by Dr. Ramesh Kumar KB, Senior Principal Scientist, KCSTE, JNTB GRI. May I invite the chairperson to kindly take charge of the session and introduce the speaker.
So a very good morning to all, and once again welcome to this plenary session of this Biospectrum 2026. As I mentioned in the introductory remarks, the conference is full of power-packed sessions with eminent scientists and academicians from different parts of the country as well as from outside the country. So in this plenary session, we are having Dr. Shijulal Nelson Sati, Scientist E1 and Coordinator Bioinformatics Facility, Rajiv Gandhi Center for Biotechnology, as well as Dr. Rajiv Ravindran Nair, Head Viral Vector Core Researcher, Kavli Institute for Systems Neuroscience, Norwegian Institute of Science and Technology. So these two plenary lectures are there in this session, and it's my proud privilege to introduce the first speaker, Dr. Shijilal Nelson Sathi. So he is Scientist 1 and coordinator of the bioinformatics facility at Rajiv Gandhi Center for Biotechnology. He is an accomplished computational biologist whose research focuses on evolutionary bioinformatics, microbial genomics, infectious disease evolution, host pathogen interactions, and antimicrobial resistance. He earned his BSc in computer science from the University of Kerala, MSc in bioinformatics from Mahatma Gandhi University, and PhD from Heinrich Heine University, Germany. Before joining RGCB, he held research positions at Heinrich Heine University, Academia Sinica in Taiwan, and the Center for Cellular and Molecular Biology, India. His contributions to evolutionary genomics and bioinformatics have earned him several prestigious recognitions, including the Young Scientist Award from KCST, the Swiss Institute of Bioinformatics International Young Bioinformatician Award, and the Carl Arnold Prize Outstanding Young Researcher Award. In addition to his research activities, he serves on the editorial board of Discover Viruses and reviews manuscripts for leading international journals, contributing significantly to advancements in computational biology and infectious disease research. So with this introduction, I invite Dr. Shijilal for the plenary talk.
Yeah, thank you very much for that detailed and kind words. So, welcome back. Good morning, all of you. Can you please make this full screen? Okay. So, currently I am heading the bioinformatics laboratory at the Rajiv Gandhi Center for Biotechnology at Thiruvananthapuram. So, maybe some of you or all of you might be knowing RGCB, which is the main campus, or the campus 1 is located in center at Pujapura. But recently we commissioned another campus in Akulam, which is near Lulu Mall actually, which looks like this, even a bit more greenish than this one at the moment. And the ground floor is basically a bioinformatics laboratory. And there is a lot of infrastructure, lot of research opportunities. I must say, the researchers, they are looking for academic activities, must consider this institute in your checklist. So what I'm going to, our laboratory is mainly working, or what I'm going to talk about is actually our research mainly. The theme of the research is on how to tackle antibiotic resistance in bacterium. So maybe now you know, when Ukraine and Russia started war, how much importance was given by the media. See, because there were a lot of live streaming is going on, and a lot of things happening here and there, lot of importance were given. And over a year, after a year, how much you see still the war is happening? Are you seeing it in the media? No. Because something happening on a daily basis, not coming as a big breaking news. Similarly, there's this slow pandemic, the evolution of resistance in bacteria is happening since long time. This is happening as a slow pandemic. And this is, because this is happening since long, and this is important thing, but it's not coming into the news as a big breaking every time. But the point is, WHO classified a list of pathogens, as you can see. Can you switch off these 3 lights if possible? So it's a critical, high priority, and moderate group actually. And also, if you look at the epidemiology of these pathogens, what is happening around the globe, And you can see this short form is escape pathogens, right? E. coli, Streptococcus, Klebsiella, and Acinetobacter baumannii, a list of bacterium. They are causing the major problems around the globe, actually. Okay. So the point is this widespread distribution of antibiotic-resistant pathogens, which highlights an urgent need for strengthened surveillance and novel therapeutic strategies. So again, the people like bioinformatics or microbiology is also a very important area to consider, actually. Okay. So, right. So the research theme basically of two major themes which is happening in the laboratory. One is understanding how this antibacterial resistant, antimicrobial resistance is happening in bacterium. Some of them are sensitive, some of them are resistant, some of them are resistant to multiple antibiotics. How does it happen? This is what we study. We use NGS technologies, comparative genomics, we use advanced phylogenomics, phylogenetics, and we try to give you answer, we try to understand. It's a highly complex mechanisms, actually, how bacterium adapting to different habitats under different conditions. What we do, we use multi-omics techniques to better understand how does this happening. Is it mutations? Is it lateral acquisitions? Bacteria can acquire things. We look into those things and try to give a better understanding of how this is happening. The second theme is actually, so first thing is you have to understand how does resistance happening in bacterium. Second thing is definitely you have to look for strategies to tackle this problem, right? So few things that we do is we look for inhibitors of efflux pumps. That's something I'm going to talk about, actually. Efflux mechanism, maybe you know the transporters, they are effluxing out all toxic material out of the cell, no? Especially the antibiotics and toxins. And that's one thing that we do. The second thing that we do is actually the environmental bacterium like Streptomyces. You know these antibiotics mostly are produced by environmental bacterium. No? Then it's the synthetic and semi-synthetic forms are derived. So we are looking for novel gene clusters which can encode novel antibacterial molecules, novel antibacterial peptides. That's something we are working on, actually. So which again is coming with a large scale genomics, metagenomics, and when it comes to prediction of antibacterial molecules or structures, it comes with a lot of structural bioinformatics approaches like molecular dynamic simulations and docking and so on. So in a nutshell, what we do at the bioinformatics laboratory of RGCB is to better understand how does antibacterial resistance is happening in bacterium, especially the escape priority pathogens. Second thing is, can we identify strategies to combat or tackle this resistance in this bacterium? That's something which I'm going to talk about. So the theme, actually the 4 PhDs and a postdoc and a project assistant, and our project manager is also around, Hari Krishna. So, if you are looking into resistance, actually, so for example, this, if you give a antibacterial molecules to E. coli or a bacterium, how does the bacterium defend it? Okay, so there are maybe you know this textbook knowledge, actually you might be knowing there are transporters actually on the cell membrane, which can pump out things. Something is coming in, it pump out, right? It's called efflux pumps. And you have a mechanisms, the antibiotic molecule coming and attacking to an enzyme, key enzyme, and the attacking site will be altered so that antibiotic stays without functioning it. So then modify the antibiotic, and the porins, actually it altered the porins so that the antibiotic molecule entered, the entry of this molecule will be restricted. And there are target site modification, target MyBus. There are 4 or 5 mechanisms are well known. It's not limited to pathogenic bacterium, but it's general, actually. Okay, so it depends, varies, and its efficiency is also varies, depends on its sensitive or resistant, actually. Okay, but one box here highlighted is an efflux pump. This is a transporter, actually, which plays a major role in drug efflux, actually. Okay, so if you are looking into this transporter, this is textbook knowledge again. You have ATP powered pumps, ion channels, and transporters, and for example, uniporters, importers, and deporters. Actually, these guys are very important when it comes to antibiotic resistance actually. So because they can pump out things, something the antibacterial molecule, antimicrobial molecules coming in can easily pump out through these pumps actually. Okay, so this membrane transport is basically mediated by distinct classes of proteins, and each with a specialized subtypes that enable the selective movement of ions and molecules across biological membranes. And when it comes to the antibiotic molecules actually, basically these antiporters are major players. And if you are again looking into gram-positive and gram-negative bacterium, again here, for example, efflux pumps. Okay, efflux pumps, again, efflux pumps belongs to a broad family of transporters actually. For example, in this case, in gram-positive bacterium, NORA or in ARCAB in E. coli plays a major role in resistance development in both the gram-positive and gram-negative system bacterial systems. And how does it work is basically an ion gradient change, whether basically that's the way it pumped out things. And most of these efflux pumps utilize these proton-coupled ion transport mechanisms to expel antibiotics actually, and thereby reducing the intracellular drug concentration and promoting resistance. So, for example, if you look into, again, what I said so far is actually, so resistance and transporters and efflux pumps. If you are looking at the efflux pumps, actually, there are different families of efflux pumps in bacterium. For example, you might be knowing this ABC superfamily, MFS, MATE, SMR, and R&D. And again, you see there is a list is mentioned here. I think it's not readable, but again, antibiotics, biocides, and dyes, and so on. And you know, these all guys can transport or expel a series of molecules actually. How a single pump can translocate different molecules is still an open question actually. It's a lot of studies are going on. But what my emphasis here is actually, in gram-positive, this MFS superfamily, okay, family of proteins, and in gram-negative, this R&D superfamilies plays a major role actually. That's what is happening. Okay, this broad substrate specificity of bacterial efflux pumps enables drug efflux and reduces efficacy of these antibacterial therapies actually. Okay, two things in gram-positive MFS and gram-negative R&D actually. So how does it look like actually? It's basically a 12 to 14 transmembrane helixes which sit in the membrane, and again similar actually is about 12 transmembrane helixes which sit in the gram-negative membrane actually. So which does this job? Mostly this does the job actually. How does this evolve? How does it become 14 transmembrane helixes or 12 helixes? That's an evolutionary question that's being studied, but I'm not going to discuss here. MFS and R&D pumps are predominantly contributes to resistance in gram-positive and gram-negative bacteria actually. Okay. So, and how this is being regulated, and you know this is a very ancient superfamily of proteins, and how does it work is basically regulated through two-component signal transduction mechanisms you might be knowing from the textbooks, and transcription regulation by local and global regulatory factors such as this MRA and repressors. And again, another thing is post-transcriptional regulation by small RNAs and regulatory RNAs and so on actually. Okay. So again, when you are talking about antibacterial molecules, not only antibiotics triggers efflux pumps actually. Okay. So for example, biocides like detergents and dyes and other dyes, it can also trigger efflux responses. And for example, this is a clear indication that, you know, exposure to biocides highly increase efflux efficiency in E. coli, but knockout of that is restored actually. Okay. So biocide induced upregulation of efflux system promoted bacterial survival, whereas loss of function abolished tolerance and restored sensitivity actually. That means the point what I'm trying to make is actually not only when you are talking about antibacterial resistance, not only resistance by the exposure of antibiotics and any toxic material such as biocides can also trigger efflux pumps actually. So for example, you know, see for example biocide exposure, and before and after you see like, you know, for example they are not so active, maybe here more active bacteria exposed to biocides undergo efflux pump overexpressions and membrane alteration, and porine reduction contributing cross resistance between biocides and antibiotics. So that means, again, I pitch the point that actually the word cross resistance. So when we talk about antibiotic resistance, you might think all these resistance develop really exposure to antibiotic continuously. That's not always the case, because they are I showed before actually. So there are multiple regulatory mechanisms actually. This can also be all be initiated or triggered by exposure to multiple factors, including heavy metals and or biocides and so on. Here is a good example of exposure to biocides like detergents or which can trigger expression of efflux pumps actually. Okay. So, for example, so there are, for example, this co-selection and co-resistance, cross-resistance, and co-regulation also occurs. That means it's not specifically to one stress condition, rather than this is happening in a co-regulator manner, actually. Okay. So, this is one of our experiment that we conducted, actually. So, in E. coli, where we looked into the expression data sets, expression profiles, under the exposure of different stress conditions like antibiotics, biocides, metals, and different environmental conditions. The question is actually, when we are talking about antibiotic resistance, you might think one or the other might triggers this one. So interestingly, you can see, for example, R&D actually, here is a efflux pumps in E. coli. That's what I said before, right? E. coli, you can see this set of genes actually, you see the darker one, which is highly expressed upon antibiotics as well as some other stress conditions, right? For example, here you can see this family of pumps are expressed slowly, I mean, this is a differential expression. That means in comparison to the sensitive strains, they are highly expressed upon antibiotics, biocides, and other environmental conditions. So what we found is actually, so for example, here also you can see in MFS, this is R&D MFS family of pumps are expressed throughout actually. You can see the little bit of darker red shade actually. That means they are expressed upon all these or most of these conditions. That means a change on stress condition can trigger MFS pumps. In turn, it can inactivate antibiotics. The point is, a bacterium can adapt to resistance not only by exposure continuously to antibiotics. Maybe continuously exposure to different biocides can also get a mechanism. It can overexpress or express its MFS pumps actually. Okay. So, for example, this is what we said: efflux pumps and regulators are upregulated across multiple stress conditions. We looked into 7 antibiotics, 10 biocides, and about 2 metals, and 4 different environmental stress conditions. Highlighting efflux pumps are key resistance mechanisms against diverse antimicrobial agents actually. Okay. So, moving further, if you are looking into, so obviously you might be thinking actually, so if you want to restore or if you wanted to block the capacity of resistance, you must attack these efflux pumps, right? So people have thought about it. There are a lot of plant-derived inhibitors for this. These pumps were already discovered actually. They were reported, and see, for example, NORA is a MFS pump, MFS pump inhibitor, and you can see NORA, MEPA is all MFS pump inhibitors actually. And there is multiple, you can also see its mode of action, and also chemical peptides are also designed actually. Okay. So efflux pumps inhibition actually, I said efflux pumps are very important, especially the MFS pumps. And efflux pump inhibition restore intracellular antibiotic accumulation. Efflux means it's pumping out, right? Once you block this entrance actually, if I close the door, you are locked inside actually. That's what you want, right? So you lock it, the pumps, so that they are synthesizing the resistant bacterium to existing antibiotics actually. Okay. So what I said so far is MFS pumps or FLEX pumps are very important. Very important. It's not only one element and it's a regulatory system of components. That's what we are working on. We call it as a model on, not one pump only. Pump is regulators and it activators, repressors, everything we call it as a model on. The whole unit, what is a whole unit that is working for the regulation of these pumps under different conditions. So we call it as model-ons. That's something we are working on. In another occasion, I will show you those details. But the point so far, what I said is these pumps are very important. Actually, you have to, if you want to restore this activity of these antibiotics, one way to tackle it is restore block these MFS pumps. Okay, so major strategies to combat drug-resistant bacterium is one way is antibiotic inhibitors actually. That's something I wanted to talk further. But if you are looking into other strategies, which I am not going to talk, is also possible. You mix this, you make these drug pair combinations or repurposing of drugs and phage-based therapies. There are different ways to do, combat AMR. But the one that we are focusing on is adjuvants. That means antibiotic adjuvants, that is FLEX pump inhibitors, can be a promising strategies to target resistance development actually. So for example, ciprofloxacin, an antibiotic which basically is a fluoroquinonal antibiotic that exert its antibacterial effect by inhibiting the DNA synthesis actually, okay, and its replication and transcription. What it goes and is binds to the gyrase. So once it's inside, what this bacteria does is this will pump out so that it cannot bind to the gyrase actually. So for example, this is what it's doing actually. So this is a pump. You have a truck inside the cell, and this is being pumped out actually. What you are planning, what you are trying to design is a lock actually. Like you are locking these doors actually. You need a key to lock it so that it can't open. If you can't open this door actually, the molecule can stay inside the cell and which can go and bind to the gyrase. So in turn, this activity and it will affect the DNA synthesis and replication machinery, and the bacteria won't be able to proceed further actually. Okay. So for example, I said there are already multiple FLEX pump inhibitors were already shown actually, already discovered. But it can also have its own side effect actually. Okay, so the FLEX pump inhibitors with a high specificity and less toxicity is still lacking actually. Okay, so that's something you wanted to pitch into further actually. Okay, so if you are asking, okay, if you want to restore the activity of antibiotics, and what you can do is you can go with an FLEX pump inhibitor. And if you are asking why a novel adjuvant is needed, is because whatever you identified till date is it does this job, but is poor pharmacokinetics and off-target toxicity and lack of target specificity is there actually. And if you wanted to further look into actually, you can't work on all the bacterium simultaneously, so you have to come up with one model. What we selected is a Staphylococcus aureus as a model because it's in fact it's a good model for a One Health setup. The bacteria can infect different animals and rodents, and also affect is also present in the environment. Is a good model. That's why we selected the SORAS to work on this EPI, FLEX pump inhibitor. And in SORAS, if you are looking into SORAS and FLEX pump families of you see ABC, MFS, MATE, and SMR. For example, if further literature suggests, mostly the MFS is doing the job actually. There are multiple members. There are 11 members. 7 of them are encoded in chromosome, and 4 of them are in the plasmid, which does this job actually. And how this works is like in opening closing the door. It's like a rocker switch mechanism, and the way it based on the proton ion gradient, the opening or closure of this pump happening. So for example, if you are looking, these pumps are whether expressed or not, you can see this NORA actually. NORA is one of the key player actually. Key player of this MFS family, and NORA is NORA is a key player, and you see they are highly expressed actually. Okay, so you see something above 0, this is a fold change, actually a fold change, and NORA was very highly expressed actually. And if you are removing this NORA or knocking out this NORA, and basically it's not resistant actually. Okay, so yeah, so reduced evolvability when exposure to EPI actually. Okay, so can I get rid of this please? Check spelling. Yeah, thanks. So for a NORA pump can be targeted to combat resistance actually, because it has been shown that NORA is highly expressed. This is not everything. It's one among the 11 pumps, but it's a key player actually. Okay, it's a major door actually, major exit, major door. So if you target one, this can be restored. It's have been shown in the in vivo models, and you can also target it. Okay. So yeah. So then if you are asking me, okay, how this is being distributed actually, so for example, first row is Staphylococcus aureus. You see all these 11 pumps are present, and these pumps are not unique things in Staphylococcus aureus. If I'm saying, okay, these MFS pumps, they are important, and if you have genomes from all these Staphylococcus species, Epidermidis, Argenteus, all different species actually, about 49 species, I looked actually how similar are your S. aureus NORA actually. I compared it to all 49. You see, this is all blast identities, and it's all about a very high 80% of or above. That means actually these pumps are not uniquely present only in S. aureus, but it's also evolved vertically. But interestingly, if you are looking into the plasmids, and you see the red ones, that means it's all the same pumps actually, or same pumps are transferred across all these 49. You might be knowing these plasmid encoded genes are highly prone to lateral gene transfer. They can exchange things, right? So what we find is actually this in MFS, there are two categories of things that are encoded within the chromosome, encoded within the plasmids. And encoded within the plasmid is highly mobile actually. It's you are giving a pen to your colleagues here actually. Okay, this is something your ancestors give you the pen actually. No? So for example, this capacity is recently circulated, and you see the regulators of these things are also highly distributed in Staphylococcus aureus. That means this regulatory mechanism is there, chromosome encoder thing is there, but some of these extra capacities are recently acquired. So our interest is mostly on NORA, but we looked into all these 7 actually. Okay. So and we have shown that okay, there are evidence of lateral transfers of some of these pumps, but our most question is on NORA. NORA is a pump, and we know the important residues. And our bioinformatics laboratory, one of the focus is actually we have a huge libraries actually, phytochemical libraries, the small molecule libraries, and different molecule libraries are available in millions of compounds. Can we select that database and screen against our NORA and identify certain testable candidates and go to the lab and validate it? This is a question that we ask actually. Okay. So if you are looking in the numbers, PubChem, maybe the bioinformatics people might be knowing, at the time of the analysis, there were about 121 million molecules. So you can imagine, if you don't use computational methods, you can't screen all these molecules. Even one third of it, or you see 121 million is a huge number. No? So it is not practical to work in the laboratory, but you can do it computational simulations actually. When it comes to these phytochemical libraries, Lotus, or in impact databases developed by the Chennai Statistical Institute actually, mostly this database of phytochemicals that are derived from or extract from medicinal plants in Western Ghats actually. So we screened all these, we considered at least PubChem and impact 2 different libraries, and we screen against this NORA actually. Previously I said NORA is a MFS pump, which is a key player in tracheoflex actually, and is high quality cryo-EM structure is available, and we used the bioinformatics techniques actually, structure-based visual screening and molecular dynamic simulations to prioritize molecules actually. We done molecular modeling, binding site identification, molecular docking, and delta G free energy calculation using MMGBSA, and we gone with the molecular dynamic simulation of 200 nanoseconds in triplicates to identify which molecule can to make stable complexes with the NORA actually. Further then we gone and looked at the its kinetics in the laboratory actually, okay, in vivo, in vitro models. So the results actually, if you are looking into, so what I am showing is the numbers here, again here, the PubChem library, and we use different filters, and filters again about, if you are again, without any screening, we removed all toxicity and interference profiling is done, and finally 2.8 million molecules were docked, and we further, this is a docking score distribution of PubChem library and impact database. It's about further, the top scoring 35,000 molecules were further analyzed from the impact means phytochemical library. About 390 molecules were further analyzed, and that means that had a very high affinity to NORA actually. So if you are looking into, these are numbers, but maybe the interesting factor here actually, our computational analysis showing about 10 molecules from PubChem that are having a very high affinity actually, which is compared to previously reported positive control. That means already known efflux pump inhibitors actually, piperine, resiperine, and etc. actually. Similarly, impact database, this one even have, you see the phytochemical molecules having even a much bigger, better scores actually, and delta G also you see very high, very good values for lower values for delta G, which is indicating their complexes are much stronger actually, bigger, better. So that means the bottom line is from our millions of molecules from both the database we select the top 10, then further look into our simulation. Simulation means you are mimicking what you are doing in the chemistry laboratory. You are putting, we can now synthetically you can make artificial simulation boxes mimicking the real world experimental setup, and then you are putting your molecule and your protein together and asking whether this can keep as a complex for longer time. That means here x-axis is actually the time, y-axis is how much it fluctuates. That means if they are a good complex, it won't fluctuate much, and if you see a lot of fluctuations, that means it's not a stable complex actually. So this one I am not showing uncommon or unstable complexes. This one all showing, you can't see the y-axis, but it's all coming within 2 angstrom, 3 angstrom distance. That means it's a signature of a stable complexes actually. Okay. Again, if you are asking me, okay, if I say there are the phytochemical, the top 10 molecule that we screened, and this is for the top 1, the top 5 actually, and this can form stable complex, you might ask, okay, what sort of bondings are made between small molecule and the protein. This is what this graph is showing. The green one showing, we can see clearly say there are hydrogen bonds actually, hydrogen bonds, and the higher the peak means they are throughout the simulation, more proportion of the time these hydrogen bonds are maintained, and they are at the key residues actually. So 5 amine compound derivatives actually, this is still work is going on. 5 amine compounds that we identified and its derivative showed a highly stable interactions with the key residues for functioning of this NORA. NORA is a rocker switch mechanism, gate opening and closure is important, and these residues are very important for that opening and closure structures actually. And we found these 5 molecules, the green ones are important connections, and boxed ones are they are connecting to these residues in the protein, which making this is very good inhibitor actually. And if you are looking at the other one actually, phytochemical molecule actually, this all pentagallaglucose, and because of its 5 rings, it's binding very nicely not only to NORA, to all 4 additional pumps actually. Again, this is showing RMSD, root mean square deviation, how much is at the whole structure level, at the residual level, also its interaction profile, which is showed actually what in a summary, in a nutshell, what we found is actually in a phytochemical database, in the previous chemical database we identified 5, still this is going on, but in a phytochemical setup actually we identified a pentagallaglucose, which is the one among the best, which is showing a highly stable interaction. Then that's a molecule that we took it further for a validation actually. This is showing its PCA and free energy landscape, which is showing this wild type versus a complex which is behaving the same, and its energy profiles are the same actually. That means they are very good complex actually. So this one we take it to the laboratory, you know, maybe the microbiology people, you know it, you know, how do you, sorry, how do you test the MICs? No? So you, for example, we test it actually, this PGG or pentacala glucose is a efflux pump inhibitor. It does not have its own antibacterial activity by its own. So you can only block this pump. That means if you are adding this along with the ciprofloxacin, this should work actually. That means, so you should give the antibiotics along and mix with the PGG. That means PGG should go and inhibit the pump so that ciprofloxacin can work actually. Sorry. So that means we tried with the different combinations, and when it comes to this FIC value less than 0.5, you might be knowing this is synergy actually. That means the effect of this one is much better than the combination of individual effects actually of ciprofloxacin and PGG. So for example, this was shown synergy actually, combination of ciprofloxacin and pentacala glucose, but in control ciprofloxacin piperine, previously known inhibitor, which is showing an additive effect actually. So what we find is previously identified molecule actually, which has a synergetic activity. PGG had a synergetic activity with ciprofloxacin when compared to the positively controlled piperine. Okay. So, and again you do this growth kinetics. For example, these are the controls actually, positive control without a blue line showing without any antibiotics or antibacterial molecule. And for example, this one piperine only, again is API, you know, you put only piperine or pentacala glucose, it does not have any activity actually, okay, because it does not have antibacterial activity by its own. The green one, you see PGG alone, it does not have it. But you know, see, but ciprofloxacin. So ciprofloxacin at this MIC level, this is a bottom line actually. Bacterium is not growing. The bottom line is ciprofloxacin 125 mg per ml. That is its MIC. But when you see ciprofloxacin 25 mg per ml plus PGG 62.5 mg per ml, which is almost similar as its MIC value. That means the point is that the amount, when you are mixing it actually, when it comes to synergy, you can reduce the ciprofloxacin amount almost 1/5 actually. Yeah? So time kill kinetics actually can reduce significantly, even 1/5 of proportion of ciprofloxacin enough to do the same MIC when you are combining with the PGG. So time kill kinetics with the PGG at a synergetic concentration reduce the bacterial growth related to the controls actually. Okay, so that's what I tried to tell you. The overall story was two-phase. One I said is the MFS pumps are important. NORA was important. We have done screening of small molecule libraries in millions. We screened these phytochemical libraries in a few thousands, and we identified certain molecules. And the one that is the pentacala glucose, you can look into the complete deal. Pentacala glucose, I talked about only the part where it's binding kinetics to NORA, but very interestingly, such a phytochemical can also simultaneously bind to other pumps, because sometimes bacteria blocking one pump activate others. So always it's challenging to find molecules simultaneously, multi-target molecules need to be found, which is a challenging task, but you know, such a large phytochemicals has a capacity to do so. So what we found is it can bind to multiple targets, and this has a very good effect, and it growth very good time kill kinetics actually in SRS, which is published actually. You can have a look if you like to read further about it. And yeah, so that's what in nutshell actually what I summarize is our bioinformatics approach to narrow down 121 to 5, and 5 I mean molecules that we are still working on it with good candidates, and phytochemicals actually. That's what we found, this PGG, pentacala glucose actually, which is a very good candidate for as a synergy molecule. But you know, again, further we are looking, this is a bulky molecule, we are looking into the small molecule analog to bypass some of its toxicity levels actually. Okay. All right, so that's the bottom line, and that's what I want to tell you. There's something which is happening on how these systems evolve, and how lateral gene transfer played a role, how these pumps evolve, and the expression, different exposure to different stress condition. I did not talk in detail, but only the point that I talk in detail is about this virtual screening. But for others, actually, those who like to know what we do, in addition to research what I mentioned, we do a lot of services actually. You can do analysis service, and we do actually internship programs, short visits. It's all possible actually. And Macfast a couple of times visited our facility. You are also welcome to do so. And please visit the RGCV website, and please visit the bioinformatics website for PhD programs and the research activities actually. And with that, I stop it actually. These are the people who does the job, and he's the whole our internal network actually. And thanks for the RGCB and funding agencies, my lab mates, my students, and thanks for inviting me, and thanks for listening to my talk. Thank you very much.
So, thank you, Dr. Shijilal, for the excellent presentation. So, this antimicrobial resistance right now we are well aware of, but how complex the mechanism behind it is, but how simply and lucidly Dr. Shijilal has explained the scene behind this antimicrobial resistance. It was a quite wonderful talk. So, if you want to have sort of interaction with Dr. Shijilal, please.
If you have a question, feel free to ask me.
So, just one question. You have mentioned around 121 million compounds you have screened. So, it is most of the compounds are synthetic as well.
Yeah. So, this is a bioinformatics resource which is hosted by NCBI. Whatever people have reported, that 121 million at the moment, at the time of analysis, it has everything. All including the derivatives of a compound, chemical entities and phytochemical molecules, or all.
That is the point.
Everything.
Otherwise, that much.
So one molecule can have an enormous number of derivatives. Right? Yes.
So that derivatives are also added.
Whatever they propose it, or where people have synthesized, everything is out there actually. So if you are, because that's a point. Even selecting a right molecule is a challenge actually. So that's why I showed you that some filters that we use, then we come up to 2.8 million. Yeah.
So among that filters, what's the role of this traditional knowledge? Is it applied in this scenario? Because these are all from foreign countries, most of these databases.
Yeah.
But we have such a rich diversity of knowledge.
Yeah.
For ages or centuries we were using.
Right.
So, for example, this Hortus malabaricus, around 740 plants, medicinal plants. So these are time-tested. Efficacy has been proved through generations. So if you concentrate on that plants, the output will be much more efficient. Do you think so?
So this is the one. Even I think so. I think your point. This is because that's something proven for centuries, right?
Yes.
That is definitely need to be taken care. But what is the challenge there is actually. So normally the most of the traditional medicines are not single molecule based. Basically it's a mixture of a cocktail of molecules. Which one is doing exactly the job is a big question actually. When we do this proteomics or metabolomics, it's very difficult to identify the concentration or even to purify things. So this is again a big research area, and there are a lot of opportunities there to characterize from the traditional knowledge what exactly the molecule which does this job actually. Because there are a lot of initiatives going on. That's all about this Ayurveda research program actually. Ayush is spending a lot of money and effort to characterize actually what are the key elements of this functional aspects of this mechanism, mechanistic aspects of this, the cocktail of molecules that are present in most of these formulations.
Yes, it is a great challenge, because recently we have compiled the phytochemicals in Cyprus. Cyprus rotunda, traditional
correct, right.
In that single plant, around 800 plus compounds have been reported from that single species itself. So
that's
it is huge, vast number of
the impact database which I showed actually, which is by a statistical institute in Chennai, they did it. So I think this knowledge, because I was telling we are rich in medicinal plants and knowledge in our system, but these need to be better utilized. Some scientific support or scientific validation required. I think these points are highly valuable actually. People should spend time on it to characterize and identify individual molecules which play a role actually. Yeah, that is.
So there is a scope for integrating this traditional knowledge into this
exactly.
with the help of
that
AI.
was the idea. That's why it took impact actually, because it's all medicinal plant derived actually. So because there is a criterion how they selected the database, they said it's mentioned that the plant should be a medicinal value. So all medicinal plant derived molecules are in that database, but this can be used for multiple purposes, not only for antibacterial screening. And if somebody can make, that's what the point, if you can make maybe a better version of it, it should be better, even better actually. Yeah, traditional knowledge I think is a must to go, one must do it actually, as my personal note actually. Yeah, it's very
so
important.
this is the relevance of this kind of conferences, because I am working in the field of pure phytochemistry. We are extracting, separating and characterizing phytochemicals, but he is on the next level, that is bioinformatics, how that information can be turned to real-time drugs. So
yeah, so
if there is any questions or
rightly said, for example, if you are looking into formulation, you might see 200 and let's say 500 molecules. All of them cannot be synthesized. Maybe once we know the structure, we can do virtual screening, and only the relevant one can be synthesized, because synthesis is a big challenge, right? So that's what the point. Okay. Yeah. Thank you.
So otherwise he will be available during the
yeah, I'm available till
break.
lunchtime. Yeah. Yeah.
So thank you very much,
Thank you.
Dr.
Thank you very
Shridilal,
much
for
listening.
this excellent session.
Thank you, sir. For the enlightening session, we now move to the plenary lecture 2. It is my privilege to welcome Dr. Rajiv Ravindran Nair, Head, Viral Vector Core and Researcher, Kaval Institute of System Neuroscience, Norwegian University of Science and Technology. He will be joining us online.
So very good morning, Dr. Rajiv Kumar Ravindran Nair. So am I audible, sir? Hello, sir.
Hello?
Yes, sir. Yes, sir. So a very good morning and welcome to this Biospectrum 2026 plenary session. So it's my pleasure to introduce you, sir. So I'm Dr. Ramesh from KCST JNTP GRI. So welcome you once again, sir. Am I audible, sir?
Yeah, I can hear you.
Okay, sir. So we are privileged to have Dr. Rajiv Kumar Ravindran Nair, a distinguished neuroscientist and biotechnology researcher serving as research project investigator and head of the viral vector core at the Kavli Institute for Systems Neurosciences, Norwegian University of Science and Technology, Norway. He earned his PhD in biotechnology from the Rajiv Gandhi Center for Biotechnology, University of Kerala. His research focuses on the development of next-generation viral vector platforms and targeted gene therapies for neurodegenerative disorders, including Alzheimer's disease and motor neuron diseases. Dr. Nair is internationally recognized for pioneering enhancer-driven adeno-associated viral vectors for precise cell-type specific gene delivery and has been granted multiple patents for his innovations. As head of the viral vector core at NTNU, he leads one of the Scandinavia's premier facilities for viral vector development, supporting biomedical research across more than 100 laboratories in 22 countries. With an impressive portfolio of research publications, patents, and international collaborations, Dr. Nair continues to make significant contributions to neuroscience, gene therapy, and translational biomedical research. So with these words, let me invite you to the plenary lecture. Sir, please.
Yeah, thank you. Can you hear me? Hello, can you hear me?
Yes, sir.
Yes, sir,
Yeah,
please.
yeah, and I have seen that some noise is coming from some other account. Maybe it's a good idea if it is mute. Yeah. So I will just start. Thank you very much, organizers, for giving me an opportunity to talk about viral vectors and their applications in life science research and gene therapy. So, being a neuroscientist, I will be focusing mostly on neuroscience applications. But these viral vectors are excellent gene delivery vehicles for other organs too. So this can be used in other organ systems for fundamental research as well as gene therapy. So I will start with a few slides on with neuroscience background. Then I will shift to viral vectors. So mammalian brain consists of different types of neurons, and these neurons in any particular brain region are different from other region in structure, in functions, in connections, electrical properties, gene expression profiles. So functionally, these the neurons in one particular brain region are different from other regions. So, for example, the neurons in visual cortex are important for vision. Neurons in motor cortex are important for movement. Neurons in hippocampus, an inner brain region, is important for memory formation. So functionally, these neurons are different from one brain region to another. So how do you identify functions? So how do you study the functions of neurons in a particular brain region? For that, we make use of in vivo neuronal activity recording. So for that, we can implant an electrode or a probe sensor into the region of our interest and record the activities of the neuron near the sensor while the animal is performing a task. So literally we can overhear what the neurons are talking to each other while the animal is performing a task. This is a very powerful technique. So you can implant this sensor probe into a brain region of our interest, for example hippocampus or entorhinal cortex, and study how the neurons in that particular brain region functions. So let's take a look at the in vivo neuronal activity recording. So here an electrode or a sensor probe was implanted into a brain region called hippocampus, which is important for the spatial memory. So the implanted animal is then allowed to explore the space inside a box, and as you will see, when the neuron near the sensor gets activated, you can see a red dot, or you can hear a popping sound. So you can see that while the animal is exploring the space, when the animal reaches a particular place in the space, that is the top left corner, a neuron near the sensor is getting activated. You can see that red dot. So it's very interesting. You can see that the neuron is silent in the rest of the space. The animal is exploring throughout the space in the box. So you can see that this particular neuron gets activated and starts firing only in this particular place, and it is silent in the rest of the space. So this particular type of neuron, which is present in hippocampus, that gets activated and starts firing at a particular place, is called a place cell, and this place cell is implicated in spatial memory. So here I have to say a few words about the institute where I am working. So the directors, my bosses, Edward Moser and Maybert Moser, they received the Nobel Prize in 2014 in physiology or medicine for the discovery of another functional type of neuron called grid cell, and this grid cell is present in a different brain region called entorhinal cortex. So here they implanted the electrode or sensor probe in a different brain region called entorhinal cortex, and they allowed the animal to explore the space inside the box. So while the animal is exploring the box, so you can see a neuron in the entorhinal cortex starts firing at multiple places. So here you can see a white dot when the neuron near the sensor is getting activated. So you can see here in entorhinal cortex a different type of neuron gets activated at multiple places and starts forming a beautiful hexagonal grid-like firing pattern. So you can see that as it develops, it forms a hexagonal grid-like pattern, like here. So you can see the hexagonal firing pattern is getting developed. So this particular type of neuron, which is present in neuronal cortex, that gets activated at multiple places and forms a beautiful hexagonal grid-like pattern, are called grid cells. They are important for spatial navigation, and they create an internal map of space inside our brain. So as I said, this particular technique, which is the in vivo neuronal activity recording, is a very powerful technique to study the functions of neurons in any particular brain region. But that is not enough. You have to do, but this is a correlational experiment. It's a correlational data. So you have to do further experiments to substantiate the causal relation between activities of neurons in a particular brain region and a specific brain function. So for that, you have to turn the neurons on or off in a particular brain region and study how that affects the neuronal activities and ultimately the behavior of the animal. So you have to literally turn the neurons on or off, or modulate the activity or manipulate the activities of the neurons in a particular brain region to establish this causality. So how do you do that? So we use molecular tools for modulating or manipulating the activities of neuron in any particular brain region. So we can introduce the genes coding foreign receptors or channels on the surface of the neurons, and then we can activate or inactivate those neurons with external stimuli like lights of different wavelength or designer drugs that can specifically bind to a foreign receptor that we can express on the surface of the neurons. So now the big question is, like, how do you deliver the genes coding these molecular tools into the cells, so here neurons, into a complex organ like mammalian brain in vivo? So you have to express these molecular tools there, and then you have to allow this animal to live for several months, several weeks to months, and then you have to record the electrical activities. So how do you do that? So here we make use of viruses for delivering these kind of molecular tools into the neurons in an intact mammalian brain. So when you hear about viruses, fears of deadly diseases rush to your mind, or unpleasant memories rush to your mind. But not all viruses are villains. Some of the viruses, like viral vectors, these are genetically engineered viruses. They are excellent gene delivery vehicles. So we can develop excellent gene delivery vehicles by harnessing the beneficial features of viruses. So nowadays we are using these viruses for fundamental research in our institute, but we are already using these kind of viral vectors for clinical practice, clinical applications for curing human diseases, and we are using these viral vectors for viral vectors as vaccines. So by making use of modern molecular biology techniques, you can remove the genes coding deleterious viral proteins that cause that make them infectious disease-causing agents, and replace them with the genes of our interest. So such genetically engineered viruses are called viral vectors. They are safe and they are non-infectious, and we can make use of these viruses to infect the host cell and produce the proteins or RNAs of our interest. So this is how the viral vector mediated gene expression works. This is a mechanism. Core protein on the surface of the virus will bind to the receptors on the surface of the host cell. So this is a host cell, and the core protein will bind to the receptors on the surface of the host cell, and upon receptor binding, the viral vector will be internalized by receptor mediated endocytosis. So the viral vector will be packaged in membrane bound endosomes, and these endosomes will be trafficked to the nucleus. So inside the cytoplasm, it will undergo pH dependent breakage, and then the capsid containing the viral genome will be trafficked to the nucleus, and it will be docked to the nuclear pore. And from the capsid, from the viral capsid, the viral genome will be will be released into the nucleus, and for some kind of viruses like lentivirus, the viral genome will be integrated into the genome of the host cell, but for some other viruses like adeno-associated virus, it will remain as episomal DNA construct. And then the viral genome will make use of transcriptional as well as the translational machinery of the cell to produce RNAs and proteins of our interest. So this is how a viral vector works. And then, so these viral vectors are excellent gene delivery vehicles for modulating the neuronal activities. So we can introduce the genes coding molecular tools for various functional activities. So various functional studies. So you can use viral vectors to introduce the genes coding GFP or red fluorescent. So GFP is a green fluorescent protein, and mCherry is a red fluorescent protein. So you can use these viral vectors to introduce the genes coding GFP or RFP or mCherry to fill the neurons and study the anatomical mapping of neuronal circuits. So when it comes to the neurons, connections are very important. So we can use these viral vectors to study the connections between the neurons, and then we can use these viral vectors to introduce foreign channels like channel rhodopsin or halorhodopsin on the surface of the neurons, and then you can just activate or inactivate. You can turn the neuron on or off using lights of different wavelengths. So for example, you can express foreign channels like channel rhodopsin on the surface of the neurons, and shine those neurons with blue light. Then you can activate or you can turn the neurons on, and you can express using viral vectors the genes coding halorhodopsin. This halorhodopsin will be expressed on the surface of the neurons, and you can just shine those neurons with green light, and you can just inactivate or turn the neurons off. Further, you can use viral vectors to express receptors, foreign receptors on the surface of the neurons, and then you can use a designer ligand that specifically bind to these foreign receptors. And these ligands will not bind to any of the endogenous receptors, so we can just specifically activate or inactivate the neurons expressing these pharmacogenetic receptors. And further, you can use viral vectors to express calcium sensors like GCAM or voltage sensors like JEDI or ASAP, and then you can monitor the activities of neurons while the animal is performing a task. So earlier I showed that you can record or capture the electrical activities, but using these kind of tools you can monitor, you can just image the activities of neurons while the animal is performing a task. So further, we can use viral vectors to manipulate the expression of an endogenous gene of our interest in cells grown in a Petri plate or organoids or an intact mammalian brain. So we can use these viral vectors to manipulate the endogenous gene expression. You can knock in or you can knock down or knock out a particular gene of your interest using viral vectors. So this is from our work, and we designed and created a viral vector that expresses microRNA that targets an endogenous gene called reelin, which is expressed in endorenal cortex. So here we created this virus that is used for knocking down or that expresses microRNA against reelin, and we injected it into the endorenal cortex, stereotaxically injected into the endorenal cortex in one hemisphere, and the contralateral hemisphere or the opposite hemisphere we injected a control virus. So you need a control virus to get the data, to analyze the data. So as you can appreciate, the expression of reelin is intact in GFP control virus expressing neuron, while there is a drastic reduction in reelin in the experimental or the reelin knockdown expressing virus. So to our big surprise, we saw, it is very interestingly, we saw that when the reelin is knocked down, when you reduce the expression of reelin, that resulted in significant reduction in different pathological forms of amyloid beta. So this injection was done in Alzheimer's disease rodent model. So if you knock down or if you reduce the expression of an endogenous gene reelin, we noticed that we reduced, or there's a significant reduction in different pathological forms of amyloid beta in this Alzheimer's disease mouse model or rodent model. So these kind of viral vectors have a huge potential to become gene therapy vectors for lowering different pathological forms of amyloid beta in Alzheimer's disease in future. So further, the viral vectors can be used in combination with different transgenic lines, and we can just we can attain cell type specific or region specific to some extent using viral vectors in combination with transgenic lines. So if you use in combination with transgenic lines, you can attain cell type specific gene expression using viral vectors. So when it comes to neuroscience, I will come back to that. Cell type specific expression is very important. So we can use viral vectors for conditional expression. So what is a conditional expression? Here the condition is like the viral vector will express a particular gene of your interest, channel rhodopsin or calcium sensor. So they will be expressed only in presence of a factor. So that is the condition here. So using these kind of conditional viral vectors, you can have a spatial as well as temporal resolution of gene expression. So that means that you can decide when and where to express a particular gene of your interest using viral vectors. So that is very it's very critical in neuroscience experiments. So this is also a work from our group. So here we designed and created a virus, a conditional viral vector that expresses a foreign channel rhodopsin, that expresses only in presence of a conditional factor TTA. So here this virus was injected into a transgenic line that expresses TTA only in a specific type of cells in entorhinal cortex. So we injected this virus into the entorhinal cortex of this transgenic line, and we use this viral vector to study the difference in connections between the neighboring regions of entorhinal cortex. So this is a very powerful tool to study the connections between the functional mapping of neural connections in that mammalian brain. So here we can decide when and where to express a particular type of gene for our experiment. So finally, this is not science fiction anymore. So we are extensively using these kind of viral vectors for neuroscience experiments, for fundamental research, but they are already in clinics for curing many human diseases. So viral vectors are excellent gene delivery vehicles to introduce genes for gene therapy applications. They are already used for as vaccines. They are also used for gene editing applications also in clinical trials. So vaccines, you all know that during pandemic, one of the main vaccine that was used in India that saved millions of lives was AstraZeneca Oxford's vaccine that is distributed under the trade name, I think, Covishield, and that is a viral vector based vaccine. So these viral vectors are already in clinics for curing diseases as well as they are used as vaccines. So now I will focus mostly on the clinical applications of viral vectors. So far I was mentioning on the fundamental research part. So mutations of certain genes make those genes non-functional. So these non-functional genes may cause genetic disorders. So you can use these viral vectors to introduce a functional copy of the faulty gene, and we can restore the function of the faulty gene. So these viral vectors can be used, are used extensively for gene therapy treatment. So they can be used for in vivo gene therapy application. So here the viral vectors are directly administered to the patients to introduce a therapeutic agents to produce a therapeutic application. So they are also used for ex vivo gene therapy application. So here the cells from the patients can be extracted. For example, stem cells can be extracted from the patient, and then you can modify these stem cells using viral vectors, and then you can expand these modified stem cells in culture, and then we can just reintroduce these modified stem cells back in patients. So these viral vectors are already used for ex vivo and in vivo gene therapy treatments. So this is a timeline overview of the approved gene therapy products that are already in markets and their delivery methods. The first gene therapy treatment that got approval was Gendicin, that was in 2004, and this was for head and neck cancer. So I have to say that viral vectors are extensively used for cancer therapy these days, for CAR T cell therapy. So since then, since 2004, several gene therapy applications using viral vectors got approval from regulatory authorities, and they are extensively used for in vivo as well as ex vivo gene therapy applications. So here are some of the gene therapy products that are used for curing human diseases. For example, Luxturna. So this is a gene therapy treatment, viral vector based gene therapy treatment for inherited retinal diseases. So here an adeno-associated virus, a type of viral vector, is used to introduce a functional copy of a RPE65 gene to restore the vision in patients suffering from retinal dystrophy that leads to blindness. So this is a life-changing treatment, and here this is another treatment which is Zolgensma. This is a gene therapy treatment, viral vector based gene therapy treatment used for treating spinal muscular atrophy, and here the viral vector is used to introduce a functional copy of SMN1 gene to the motor neurons, the affected motor neurons in spinal cord, and then we can restore the function of the faulty gene. So the spinal muscular atrophy is a devastating disease that's a muscular disorder, neuromuscular disorder, and that eventually that leads to difficulty in movement, swallowing and breathing. So this is a life-saving treatment. So recently one gene editing therapy that uses CRISPR-Cas got approval from regulatory authorities, and along with that two other gene therapy treatment using viral vectors also got approval for severe sickle cell diseases and beta thalassemia. So the first one is Lyfgenia, that is used for treating, it's a viral vector based gene therapy treatment for treating sickle cell diseases, and then Sindaglo is another viral vector based gene therapy treatment for treating beta thalassemia. And then this is a very interesting, promising clinical trial, and this gave very promising results for Huntington's disease. So here this is a viral vector based gene therapy treatment that silences a mutant Huntington gene that causes Huntington's disease. So this is a viral vector based gene therapy treatment under clinical trial, but this is giving very promising results. So these are a handful of gene therapy treatment that uses viral vectors, and all these treatment were possible because of the application of viral vectors. So here the viral vectors are used to introduce a therapeutic agent to cure the disease or halt the disease progression. So not only that, this is the future. So around 238 clinical trials, but now the list has gone up. Around 300 clinical trials are already in pipeline that uses one type of viral vector, that is AAV, and this is for treating a broad spectrum of major human diseases like cancer, neuromuscular diseases, metabolic diseases, ocular diseases, cardiovascular diseases, etc. So this is the future. So coming back to the production of AAVs, production of viral vectors in lab, the production scheme is similar for most of the viruses. So this is the production scheme. So here we use mammalian cells to produce the viral vectors, so the mammalian cells are used as factories for producing the viral vectors. So all the genetic information that carries the essential elements to produce the viral vectors are provided in the form of plasmids. So these plasmids carrying the transgene, the gene of our interest, along with all the helper genes, are introduced into the mammalian cells in culture. And once the cell has all the genetic information, then the cells will act as the factories, and it will produce the virus, and it will be secreted into the medium, or some of the viruses will remain inside the transfected cells. So this process is called transfection, and you can collect the medium, and you can just collect the cells filled with viral particles, and then you can lyse open the cells, and then you can just purify the viral vectors using chromatographic techniques, or you can use ultracentrifugation techniques. So finally, the viral vectors that we produce can be used for fundamental research, as we are doing in our institute. They can be used for gene therapy treatments, as some of the examples that I mentioned in the previous slide. They can be used for vaccines, as vaccines also, for saving millions of lives. So the viral vectors are having several advantages, but they have significant disadvantages too. So they have several advantages, like they leverage millions of years of evolutionary adaptations of viruses, so they can just enter easily into a host cell, and then it leads to efficient gene expression and delivery. And we can just using genetic engineering, you can just engineer the capsid, the code proteins on the surface of the viruses, and then you can just create cell type specific or region specific viruses or viral vectors. And then some of the viruses can be used for long-term gene expression. For example, lentivirus, they integrate into the genome of the host cell, and they are already established, as I mentioned. They are already used in curing human diseases. So we have a very strong foundation of knowledge and history and safety data. And some of the viruses can be used for high payloads, like high cargoes. So big cargoes can be integrated into or packaged in some of the viral vectors. So what are the drawbacks or disadvantages? Some of the viruses, or most of the viruses I would say, have immune response. So multiple injections can provoke immune response, and this may reduce the efficacy, as well as it will pose risk to the patients. And some of the viruses, like lentiviruses, I mentioned, they integrate into the genome of the host cell, so that will disrupt some of the normal genes, and that can activate oncogenes, and which can lead to cancer or mutations. So there are nowadays ways to get around this drawback, but this is also a concern. So producing viral vectors, we produce hundreds of different types of viral vectors in our facility, but viral vectors that can be used for clinical applications, human clinical applications, is technically very challenging. It's very costly, and it's very time consuming. And for some of the viral vectors like AAVs, the cargo capacity is very small, so that is also one of the limitations. So the last part, I will switch gears to what is happening in my lab these days. So we are currently focusing on tailor-made AAVs, or adeno-associated virus. So that's a type of viral vector for cell type or region-specific gene expression. So when it comes to the neuroscience experiment, it's very critical that we have cell type specific or region specific gene expression. This is because any leaky expression or any side effects in other type of cells or in other brain regions can cause serious side effects that has serious consequences. So when it comes to the mammalian brain, there are hundreds of different types of neurons, and these neurons, as I said, they differ in structure and function, gene expression profiles. So how do you target specific neurons in a complex organ like mammalian brain? So we can make use of the intact tropisms of some of these viral vectors. So some of these viral vectors have specific cell type specificity, so we can make use of such viral vectors. Or you can use genetic engineering. You can use genetic engineering to modify or create new viral capsids with specific features, with specific cell type specificity, or we can use cell type specific promoters or enhancers for cell specificity. So in our lab, we are using enhancers for cell type specificity. We use enhancers as drivers for cell type specific gene expression. So what are enhancers? So enhancers are regions in the chromosomes where the transcription factors or activators can bind, and thereby they can activate or enhance the expression of a gene. So when it comes to, for example, in embryonic development, the same gene will be expressed in different regions. So for example, the same gene will be expressed in brain, the same gene will be expressed in limb, but they are under the tight control of different cell type specific enhancers. So for example, the gene X is under the tight control of brain specific enhancers in brain. So the transcription factors, brain specific transcription factors will bind to brain specific enhancers, and that will enhance the expression of the gene X. The same gene will be under the tight control of limb specific enhancers in limb. So here the transcription factors or activators will bind to limb specific enhancers, and that will enhance the expression of gene X. So if you identify cell type specific enhancers by molecular biology techniques or biochemistry techniques, you can use these enhancers as a locan key or a code for cell type specific gene expression. So the region specific enhancers can be used can be used to drive region specific or cell type specific expression of foreign genes. So in our institute, Stefan used he's micro dissected different brain regions. This is the brain section from rat. So he micro dissected different brain region, and using ChIP-seq analysis. So these are molecular biology or biochemistry analysis, and then followed by bioinformatic analysis, they identified region specific or cell type specific enhancers, and they demonstrated that if you use these cell type specific or region specific enhancers, we can create a cell type specific transgenic lines, transgenic mice lines. And then in my lab, we validated some of these enhancers. We used some of these validated enhancers, and we put these enhancers into viral vectors and use these viral vectors to drive the expression of foreign genes like green fluorescent protein or red fluorescent protein or other cargo genes. And we demonstrated that if we use cell type specific enhancers, we can drive cell type specific or region specific enhancers. So for that, we got a patent for this technology, and we call this enhancer driven gene expressing adeno associated viruses or EDGE AAVs. So we got a patent, and we already published this data. So this is very relevant because these kind of EDGE or enhancer driven AAVs can be used as gene therapy vectors, cell type specific gene therapy vectors in future for several neurodegenerative diseases. So this is a slide to show the specificity of EDGE AAVs or enhancer driven AAVs. So here we integrated one of the validated enhancer that is very specific to some of the cells in entorhinal cortex. So remember, this entorhinal cortex is the region that is that contains the grid cells I mentioned in the beginning. So we use this enhancer specific, the enhancer which is very specific to some of the cells in entorhinal cortex, and we designed and created a virus that expresses green fluorescent protein. So this green fluorescent protein, so here this is yellow, and this is a brain section from rat, and all these red dots are neurons. So this is a very thin section which is 50 micron thick section. So you can see the expression. We injected a ton of virus into this brain region in vivo, and we allowed the animal to survive for almost a month, and after 1 month we sacrifice the animal, and this is the brain section that we analyzed. And you can see, even though we injected a lot of virus in this brain region, this virus expresses only in specific cells in a layer. So in brain cortex, the neurons are arranged in layers. So this virus expresses only in a particular layer, layer 2 of entorhinal cortex, and it even spared a different type of neurons in the same layer. So these type of viruses can be used for cell type specific or region specific gene expression. So what is the relevance of these kind of cell type specific or region specific enhancer driven or edge AAVs? So when it comes to neurodegenerative diseases, the pathology starts at a particular brain region, even at some particular cell types, and from there it progresses into in a characteristic propagation pattern as the disease progresses. So this is the case for Alzheimer's disease. So pathology, one of the pathologies, tau pathology, starts at transentorhinal cortex and in an early Alzheimer's disease patient, and from there it progresses into the higher order brain regions, the higher order cortical regions, and then it spreads all over the brain and damages the brain. So this is the case for Parkinson's disease also. The pathology starts at a particular brain region in substantia nigra pars combatta, and from there it progresses into higher brain order regions. So this is a brain section from an early Alzheimer's disease patient, human patient. So you can, this is a brain section, and this is the transentorhinal cortex. The tau pathology here is shown as these dark spots, starts at the transentorhinal cortex, and from here it progresses in a characteristic propagation pattern into other brain regions at a later stage, damaging the brain. So here you can see that we have a viral vector that is very specific to a brain region that is layer 2 of entorhinal cortex, and this is a region which is equivalent to transentorhinal cortex in human patients. So using these kind of viral vectors in future, we can target starter cells at the epicenter of these kind of diseases like Alzheimer's disease. So you can package therapeutic genes using these kind of viral vectors and deliver these viral vectors so that they can target these starter cells at the epicenter of these diseases, and we can hold the disease progression, or you can just cure these kind of diseases, deadly diseases like Alzheimer's disease. So this is what currently happening in our lab. We are developing. We have right now we are fine tuning. We already got a patent on this. So we are developing a targeting mechanism that uses enhancers that can target payloads, genetic cargoes that can lower the disease progression, that can lower the pathology in the starter cells at the epicenter of these kind of diseases. And this is where this has a lot of benefits that can increase the efficacy and specificity of the treatment, and this is very safe because it is addressing only the epicenter of the disease. As I said, a side effect or an adverse effect in brain is very detrimental. It can cause serious consequences, and this reduces side effects. These kind of tools reduces the side effect. So this is my last slide. So this take-home messages. So viruses are very efficient for in vivo. There are other methods I have to say, like lipid nanoparticles, and there are exosomes, extracellular vesicles, but viral vectors are very efficient so far for in vivo gene delivery, and they are used for introducing molecular tools for fundamental research in intact organs, like complicated organs like mammalian brain, and this is extensively used for fundamental brain science research and other life science research, and they are used for gene therapy applications. And this is something that I haven't mentioned in my presentation, but viral vectors are already used in clinical trials for gene editing therapies. So here they are using CRISPR-Cas. So CRISPR-Cas based gene editing therapies are also booming. They are also the future. So here they are used. They use viral vectors to introduce the components of CRISPR-Cas, but challenges remain for their in vivo clinical gene editing application. So this is still under clinical trials. So novel viral vectors, we are developing novel viral vectors like enhancer driven gene expression viruses like edge AAVs, as I mentioned. They can deliver therapeutic genes to specific cells within intact organs like brain. So that's all in my presentation, and thank you very much for my funding sources, and thanks to our directors, member Edward and Biane, and thanks to my lab members. And if you have any questions, and depending upon how the format is, like I'm happy to take some questions. But if you have any questions beyond this presentation, you can definitely contact me by social media apps. Yeah.
Thank you, Dr. Rajiv Ravindran Nair, for the very insightful, informative, and elaborative talk on viral vector-based gene delivery systems. So, the functioning of brain is always a quite fascinating question for the humankind, and a very basic intellectual enigma. So whatever being done on to resolve this enigma is quite interesting. So the nuances of neuron were elaborated in a lucid manner, especially on the viral vector-based gene delivery systems. So it was quite an interesting session, and thank you. And if there is any questions or interactions from the audience side, please.
Good afternoon, sir.
Sir, are you audible, sir?
I can hear you. Yes, please.
Sir, I just want to ask that, sir, can we use viral vectors as delivery system for therapeutic proteins or genes across blood-brain barrier instead of using invasive surgeries or injections? Is it possible to deliver these viral vectors across the blood-brain barrier?
Yeah, that's an excellent question, because of time limit, I haven't mentioned. We definitely we are making several of these kind of viral vectors. So you can change. There is a capsid around this the virus, and we can change the capsid proteins, and we can just have specific tropisms. And one of the viral vector that we use is AAV PHPEB. It's a mouthful, but this is a viral vector that has a characteristic feature that it can cross the BBB. So you can use these viral vectors to inject systemically, and you can inject. We are extensively using this in animal experiments. We can do a tailvein injection or retroorbital injection, and then these viruses can cross the BBB. But the only thing is that if you don't use these kind of cell type specific viruses, it can infect all over the brain. It will be a global expression. So that is one of the reasons why we are choosing invasive expression. Then it is more local. But yes, we already have specific viral vectors that can cross BBB. The only drawback is that if you are using it for human, we need a ton of virus, because you need to cross the BBB, and these viral vectors will be crossing other organs too. But yeah, that's a long answer, but of course we have.
Yes, sir. Thank you,
Such
sir.
viral vectors. Yeah.
Is there any other questions?
Yeah, please go ahead.
Good afternoon, sir.
Yes.
Okay, so my doubt was, after injecting the viral vector, how long it will take for expression? Like, if it's expressed, is it like permanent, or do we need to undergo any repeated treatment procedures?
Yeah, that's a good question also. Yeah, that's a very good question, and it depends upon some of the viruses. For example, lentivirus, if you are using, that integrates into the genome of the host cell. So that is used for long-term gene expression, that integrates. But the drawback is that then it can integrate at the random site, and so I'm talking mostly about the clinical site. But these kind of viral vectors are already using for clinical practices for stem cell modulation. So yes, we can use these viral vectors for long-term gene expression. Some of the viral vectors can stay as episomal DNA construct. So if it is a rapidly dividing cell population, like in liver, so those kind of viral vectors is not the right choice. But when it comes to the brain, it's okay, because the neurons don't divide. They are post-mitotic after development. So in that case, AAVs are totally fine, because they can remain as episomal DNA construct, and it will not get diluted as the cells divide. So neurons don't divide. And in our experiments, I don't have time, but I have some of the slides where after injections we sacrifice the animals after 1 year, after several months. The expression is fantastic. You can see beautiful expression there. So for even with some of the viruses with that doesn't integrate, you can have long-term gene expression. And this is already reported in muscle disorders, where you inject viruses, and then that can last for several years. And if the expression goes down, probably you have to inject another dose for humans. But I think this is this is possible.
Thank you, sir.
Yeah.
Sir, one more question.
Yeah, go ahead, please.
So what challenges are facing while recording the neural signals, and are the results reproducible?
Sorry, I didn't understand. It's a bit noisy, but can you repeat the question, please?
Sir, what challenges are facing during the recording of the neuro signals, as well as are the results reproducible?
Yeah, I didn't quite get it, but this kind of recording is something which is a well-established technique. So you can implant these kind of I'm just going to the slide to explain it. So these kind of techniques are already there for several decades. So probably you can see. So you can implant these kind of sensors or electrodes, we call, or probes into a particular brain region of your interest, and then you can do this kind of electrophysiology studies, and this is definitely reproducible by different labs across the world. And this place cell discovery was done several decades, and the grid cell discovery was done I think it's in 2006, around 2006, that time period. But several labs are doing these kind of experiments. But nowadays, the big change now is like initially when they got the Nobel Prize, John O'Keefe for place cell and Moses for grid cells, they could record from a handful of cells, like 4-5 neurons from a particular brain region. But with the neuropixel, this is a new advancement, recent advancement. We can record from thousands of neurons from an intact brain region. So we can just right now study how the neurons are talking to each other. So that's a big advancement. And other than that, there is a microscope. We can just put it on the brain over the skull, and you can image the neurons right now while the animal is doing a task. So that's also a long answer. So these kind of electrophysiology techniques are age-old techniques, and they are well established, and they are done by several labs, and this is extremely reproducible. Yes. But it's very the infrastructure is very demanding. It's very challenging it needs special infrastructure to do these kind of experiments. That I agree. Yes. Go ahead, please.
So thank you, sir, for the very interesting and informative lecture. Also, it was quite interactive as well. So thank you very much, sir.
Thank you very much.
Thank you, sir, for sharing your expertise and valuable research insights. I would also like to thank Dr. Ramesh Kumar KB for chairing the sessions. This concludes the morning session. We will now break for lunch, which has been arranged in the college lobby. And the afternoon session will start promptly at 1:45 p.m. The college lobby is at the ground floor of this building.
Hi, Tina. I'm not sure if you can hear me.
Recording in progress.
Hello, Teresa. Teresa.
Dr. Ranjit. Ranjit.
Hello.
It's Ramesh from TBGRI. So we are waiting for the speaker. Just a few minutes.
The speaker was here actually. He joined and he left.
Okay. So we are connecting to him.
Good afternoon and welcome back. The invited lecture session will be chaired by Dr. Ranjit Thomas, FRCS, Associate Professor, Department of Chemistry, SB College, Changanasheri. He has been awarded the prestigious designation of Chartered Chemist. May I invite the chairperson to preside over the session, who will be joining us online. Now, I invite Professor Ekne Emmanuel Wike, University of Witwatersrand, Johannesburg, South Africa, to deliver the second invited lecture. I request Dr. Ranjit Kumar to introduce the speaker.
Opening. Good afternoon, distinguished dignitaries, eminent scientists, respected faculty members, research scholars, students, and dear participants. Am I audible to you?
Yes, sir.
Thank you. It's a great privilege to chair this invited lecture session of Biospectrum 2026, International Conference on Recent Advances in Biological Sciences, Bridging Research to Breakthroughs, hosted by Macfast College Tiruvalla. The central spirit of the conference is both timely and scientifically compelling to examine how contemporary biological sciences are moving from descriptive understanding to mechanistic insights, from disciplinary specialization to integrated platforms, and from laboratory discovery to translational impact. In an era shaped by genomics, proteomics, metabolomics, bioinformatics, biomarker science, biosciences, and data-driven biology, the phrase putting research to breakthrough is not merely a conference theme, and is a call to rethink how biological knowledge is generated, integrated, validated, and applied for human ecological and societal well-being. The invited lecture sessions brings together two distinguished scholars whose work represents some of the most exciting directions in contemporary biological sciences: integrative omics, biological data science, biomarker discovery, molecular oncology, and precision medicine. It's my honor to chair this session and to introduce the eminent speakers. The present session is intellectually significant because it brings into dialogue to strongly complementary scientific domains. On the one hand, we have integrative omics and bioinformatics, while large-scale biological data are transformed into meaningful molecular gaps, disease signatures, and predictive frameworks. On the other hand, we have translational cancer biology and precision medicine, where molecular mechanisms, biomarkers, and population specific disease patterns are investigated with the aim of improving diagnosis, prognosis, and therapy. Together, these lectures represent the direction in which 21st century bioscience is rapidly moving, from molecules to science systems, and from data to interpretation, and from mechanisms to clinical relevance, and from global knowledge to context-sensitive health solutions. The first section speaker of the day is Professor Ekene Emmanuel Wike. It's now my honor to introduce our speaker, who is Associate Professor in the Department of Surgery at University of Witwatersrand, Johannesburg, South Africa. Professor Wike obtained his MSc with distinction in molecular and cell biology and later completed his PhD in the University of Witwatersrand. And his research is located in important of molecular oncology, biomarker discovery, cancer biology, multi-omics, and precision medicine. Professor Wike's research focuses on discovery of biomarkers and the elucidation of molecular mechanisms involved in development and progression of hepatobiliary cancers, as particularly among the patients of African ancestry. This is a scientifically and socially important area because cancer biology must increasingly account not only for the universal molecular mechanisms, but also for population-specific genetic, environmental, and clinical contexts. His use of multi-omics approaches places his work within one of the most advanced frontiers in translational cancer research. He is also deeply interested in applying precision medicine to improve cancer diagnosis and therapy in Africa. This aspect of his work is specifically significant because precision medicine must become globally inclusive. It should also remain confined to not to too few specific population databases and healthcare systems. Professor Wike contributes to a broader, more equitable, and scientifically robust model of cancer research. He has received several awards and fellowships, including 2023 NRF Research Excellence Award for Emerging and Early Career Researchers and NRF Excellence Fund Medical Research Council Fellowship for 24-25. He is actively involved in postgraduate supervision at the master's and doctoral levels and contributes to several national and international committees. His words speaks directly to the future of biological science, a future in which molecular understanding is very important to give precision medicine. Now I warmly invite Professor Ekene Emmanuel Navake to deliver his invited lecture. Welcome, dear Professor Navake.
Thank you so much, Dr. Thompson. Can you see my screen?
Yes, sir.
Yes.
Okay. And you can hear me?
Yes. Perfect.
Okay. All right. Thank you so much. So again, I just want to thank the organizer of this wonderful conference for this opportunity to present. I really appreciate all the organizers, including Dr. Merlin. Thank you so much for the opportunity to present. So today I want to talk about our work that we're doing on multi-omics of pancreatic cancer. We are doing some of this work in a range of cancers, including biliary cancer, breast cancer, but today I want to focus on pancreatic cancer and some of what we've learned over the years in African patients. So I have no disclosures to make. And so pancreatic cancer is an aggressive and very lethal cancer. It has a very poor prognosis. It has a 5-year survival of about 13%. In comparison to other different cancers, you have 5-year survival of over 50% in some cancers, but in pancreatic cancer, it's still at 13%, which is very low. And it's already surpassed breast cancer in many nations worldwide as the third leading cause of cancer death. Sadly, a lot of patients, 1 in 4 patients will die in the first month of diagnosis, and we know 9 in 10 patients will not live up to 5 years. So it's a very aggressive and devastating cancer. In South Africa and in many parts of the world, it's the incidence and mortality is rapidly increasing. Like I said, it's one of the leading causes of cancer-related deaths. And we find a lot of issues and mortality in Black Africans. Even in America, we have African Americans, for example, having the highest mortalities. And this is similar to countries like in the UK, where we have multiracial societies. And in general, we still know that this cancer is grossly underreported, and I will mention why it is. So it's a very devastating and lethal cancer. And like I said, in a few years, we're going to see the statistics double. Again, this is just projecting for South Africa, but for many countries all over the world, we are projecting that this cancer, the incidence and the mortality will double in the next decade. Now, the main problem here is the fact that there's late detection. So this cancer, in most cases, it's diagnosed in the late stages when this cancer has spread to other organs. And this is mainly because the pancreas obviously is hidden within the stomach. So a lot of the symptoms that you find are actually symptoms that can be attributed to other diseases. So for example, you can have a stomach bug, you can have diarrhea, for example, or there's no specific symptom. And so in most cases, a lot of patients actually present when it's quite late and when this cancer has spread. So that's a huge problem. And like I mentioned earlier, the second issue that we have is the aggressiveness. It's a very aggressive cancer. You see patients coming to the clinic and within a few months, they're already dead because it's very aggressive, which again, we'll touch on that, but it has a lot to do with the biology of the cancer. And finally is the treatment. So I know some of you are just the recent ASCO meeting where a drug targeting KRAS finally was able to double the survival of pancreatic cancer after so many years of ineffective treatment. Even immunotherapy treatment wouldn't work for pancreatic cancer. So what that drug did was at least give us a bit of hope that it is possible to still target pancreatic cancer. But by and large, we still have most of the treatment patterns that we have are ineffective towards pancreatic cancer. And hopefully with this new drug and hopefully more new drugs to come, we're hoping that we can be able to target this cancer. So for us, we think that the solution would be to identify new biomarkers. For example, in order to avoid the late detection, we need more sensitive and specific markers that could identify pancreatic cancer early, because several studies have shown that if you are able to pick up this cancer very early, at least you are able to intervene, maybe through surgery, to cut out the tumor, and hopefully the patient survives much more longer. So we need new biomarkers and new targets to be able to identify and track this disease over time. We also need to identify risk factors. So there's a lot of risk factors, whether it's diabetes, obesity, that are well known. Again, but we need more of those risk factors that are able to be highly sensitive and specific enough. So maybe we can be able to screen specific populations of patients. And lastly, we need a better understanding of this disease. And there's a lot of underlying molecular mechanisms that we need in different populations. So whether they're Asian population, because a lot of the or African population, a lot of the information that we know about these cancers, especially pancreatic cancer, are from European populations. So we need to do more of research in our own local population to be able to identify some of the mechanism. Why is this cancer very aggressive? Why is it ineffective to treatment? So we need to understand this disease better. So what we've done over the years is, like I said, in different cancers, including pancreatic cancer, is that we've taken more of a multi-omics approach. So what we do, for example, is when the patient comes into the clinic, we're able to take blood samples, we take tissue samples, and in some cases, and I'll touch on some of the studies, we take saliva biopsies, we even take stools, for example, and then we are able to obviously take a full clinical profile of the data, including demographic data and clinical data from those patients. And the whole point is to look at these different samples on different levels of the omics level. So we're doing genomics, we're doing epigenomics, we're doing transcriptomics, proteomics, metabolomics, even metagenomics, where we'll be looking at some of the bacteria involved. But we look at all these different omics, and then finally we are able to integrate this data properly. Because what our hypothesis is that when we are able to look at these different levels together, we are able to identify more clinically relevant biomarkers, more biologically realistic mechanisms, and obviously we reduce false positives. The whole point is obviously to identify biomarkers, to identify risk factors, and to able to understand this disease properly within our population. Instead of looking at single omics level, we're looking at multi-omics, and we are combining it all together. And I will share some of the work that we've done so far to do this. But this is the approach that we've taken in general in a lot of our cancers of patients coming to our hospitals. So just going into some of the work we've done, I did mention that we did metabolomic profiling, and one of the first studies that we did, and this was published in 2021, and when we just started this whole multi-omics thing, was on metabolomics. And again, from pancreatic cancer, for example, we take samples from the cancer patients, but also we also take controls, and controls are very important for us. We get healthy individuals who volunteer their blood samples to be able to, obviously compare what we are looking at, but we also get benign conditions such as chronic pancreatitis. We know chronic pancreatitis, for example, is a risk factor for the development of pancreatic cancer. So we also take samples from healthy individuals, but also from benign individuals who are not cancerous. And then we take from pancreatic cancer patients. As you can see on my slide, we take samples from pancreatic cancer patients from different stages. So we have the resectable patients where the tumor is still localized in the pancreas, and they're able to still resect the tumor from the pancreas. So, patients who are resectable, where the tumor is still localized, we can still perform surgery on those patients. So we take those patients, also we collect the samples, and then we have locally advanced, where the tumor is no longer, is still within the pancreas, but it has spread to other parts of the pancreas. And then we have a group of patients which is the most devastating and more aggressive, where the tumor has spread to other parts of the body. So these all have pancreatic cancer, but at different stages, and we take their sample. And one of the reasons we do that is to be able to monitor the expression of whatever we find across the different stages. So we're not only looking for diagnostic biomarkers, hopefully, but also prognostic biomarkers. And like I said, this is one of the first studies that we did. And we found some things quite interesting. So we found that some specific metabolites are obviously significantly dysregulated across the patient group. So, for example, we found that lactate, glycine, ascorbate, and pyruvate were significantly dysregulated. So we found that, for example, as the patients become more severe from healthy control to metastatic, we found that, for example, lactate begins to increase. Same thing with glycine. On the other hand, if we found that as the patient become more severe from healthy participant to metastatic, we find that the levels of ascorbate begin to decrease, right? So if you look at these metabolites, we know that metabolites, for example, lactate, for example, is linked to inflammation, for example. So we found that metabolites linked to inflammation increase as the disease progresses. And the reverse is the case. For example, we know ascorbate, which is well known and has been used to target different kinds of cancers, vitamin C. As the cancer becomes more deadly or becomes more severe, the levels of ascorbate begin to decrease. So we found this in our patient, and we thought this was very interesting. But one thing that we found is that inflammation seems to be a key driver of this cancer across our patient population. So this was quite interesting for us. And like I said, when we talked about inflammation, we looked specifically at the GPS score, which is used as an inflammation marker. We also looked at glyc A and glyc B, which are well-known metabolomic inflammatory markers. And again, the common thread is that as the disease becomes more severe, we are seeing inflammation increasing across board. And that begins to, the question of the role of inflammation begins to come in this kind of cancer. So this was one of the first interesting findings that we found within our patient population. So the next thing that we actually looked at was the immunomics. We looked at immune levels, for example. We did a lot of immunophenotyping. We did a lot of real-time PCR, ELISA assays, and we were able to come up with a phenotypic profile of our patient populations, and we found very key findings. We also did a Ross assay, looking at the oxidative stress levels within our sample population. And again, the common thread is that as the disease progresses, we see an increase of ROS compared to healthy participants. Specifically, we looked at the immune cells. Like I said, we found that effector immune cells could be essential in predicting prognosis on our PDAC population. We found that increased granulocytes and neutrophil levels and decreased T lymphocyte levels are associated with better outcomes. So we were able to come up with our profile, the immune profile of our patient population, and we were able to link them to specific outcomes. So for example, like I said, decreased T lymphocyte levels were linked to better outcomes. And these were all the work that we've done within our population when it comes to immune profiling. An interesting work that we also looked at, and this was a PhD project of Dr. Angela, was the microbiome. We all know that the role of microbiome within cancer, within a lot of diseases. And in pancreatic cancer, we were able to investigate within our sample, within our population, the microbiome profile of our work, of our patients. So here we collected, obviously we had control participants who donated some of their samples, and we had pancreatic cancer patient also. So from there, from each of those patients, we got samples from the mouth, from the stomach, from the duodenum, from the bile, and the pancreas. And one of the first things that we found is that the microbiome communities at the different sites were different. So, the microbial community in the mouth is slightly different from what we found in the stomach or the duodenum and the bile and the pancreas, which is understandable. But as you can see on the screen, you can also see slight differences between the pancreas and the patient with pancreatic cancer and those without pancreatic cancer. But what was interesting for us in this microbiome analysis was that, when you compare the communities in the pancreatic cancer and the control samples and control patient, we found a group of obviously microbiota that have been linked in inflammation and immune modulation. So what we're seeing again, so whether the Pseudomonas, Acinetobacter, the Geobacillus, and the Corynebacterium, as you can see that there is a significant difference between this group of microbiota in pancreatic cancer patients and the control. And again, these microbiome communities are linked very much to inflammation. So if you can see, we are seems to see a common trend of inflammation across pancreatic cancer patients compared to our control participants. We just finished the work now, which is impressed on proteomics analysis. And again, we're able to compare healthy control with chronic pancreatitis and with pancreatic cancer at different stages of the cancer. And again, we use a mass spec analysis looking at extracellular vesicle in plasma samples. And again, one of the things that we find in more and more is that we've now identified a group of proteins that could be used as a diagnostic and prognostic marker, and we are currently validating those protein biomarkers now in a bigger population of patients. But what's important for us here also, that if you look at dysregulated proteins and the key proteins that are very much and significantly dysregulated, these are proteins that are linked to inflammatory processing. So again, we're looking at different omics levels, inflammation seems to be the key thing in pancreatic cancer patients. Again, I won't go into more details here. In whole exome sequencing, we also did some whole exome sequencing, and what you can see there by the screen is some of the differential expressed mutations, and again, some of the key genes that we find to be highly mutated across some of our samples, tumor patient population, are those very much informed in inflammation, inflammatory processes. So I think taking together what we've found so far across the different omics that we've done over the years is inflammation. There seems to be a bidirectional role that inflammation plays in immune dysregulation, in microbiome dysbiosis, in stroma remodeling, and in metabolic reprogramming. So we see inflammation being central in so many of these processes, and we are beginning to hone now into inflammation as key into the development of pancreatic cancer. And just talking about inflammation of pancreatic cancer, we know that if you're looking at normal pancreas all the way down to pancreatic cancer tissues, we find that actually there's a lot of inflammation that happens. And this inflammation results to a lot of, whether it's genetic mutations or whether it's immune dysregulation or microbiome dysregulation, we now see that inflammation is key from progressing from normal pancreas to the tumor, to pancreatic cancer. So for us going forward, I think one of the key questions that we want to ask, and multi-omics have helped us to actually identify this inflammation as a major process in pancreatic cancer. We want to know what triggers inflammation in pancreatic cancer. I think that would be important for us going forward. What are the key things that trigger inflammation? Now, can you now effectively monitor that inflammation from normal to pancreas? And this is where we begin to hopefully be able to identify some key proteins or metabolites or whatever it is that can help us distinguish pancreatic cancer at early stages. But also importantly, can we target inflammatory processes in pancreatic cancer? And if you target an inflammatory process, are you able to actually stop the progression of pancreatic cancer? So I think these are the key questions for us going forward. We've started some work quite recently on targeting inflammation in pancreatic cancer. We just published this, and what we found, for example, is that the complement pathway actually plays a key role in pancreatic cancer, and when you target that, you are able to actually attune the cells to either become less proliferative or more proliferative. And so I think going forward, this is also a pathway that we're looking at very specifically within the inflammation and tumor progression pathway in pancreatic cancer. And so what we've done so far so good is use multi-omics to be able to establish the role of, or just look at tumor progression in pancreas within our patient population, and integrating these different multi-omics layers have actually shined more light on the disease in our patient population. Okay, so just to acknowledge a few people, we have lots of people, especially the patients. So lots of patients have been involved in this and they've freely given their samples for research. So we thank them a lot. So this is really a lot of effort from different groups of people, from surgeons to scientists to bioinformaticians to admin people, to especially the patients. So we thank them, and to our several grant funding organizations. So thank you. I'll be happy to take any questions. Thanks.
Thank you very much, Person Avaki. If any of the audience have got any questions to ask, he's willing to ask and give answers. So, any questions, please, audience can ask.
Sir, I would like to ask a question.
Yeah, please ask.
Yes, sir. Sir, how do we identify that this pancreatic inflammation is caused by cancer itself, other than any other disease condition or like a blocked bile duct? How can we make sure that this inflammation is caused by cancer tumor?
So that's a very good question, and I think what is important here is that what we do, like I said, is that we are able to take control samples. So not just people that are healthy, but also people with benign conditions. So within our control population of patients, we also have those that have chronic pancreatitis. We also have those who have different benign pathologies, including those who had blocked bile duct. So we have, if you, for example, this proteomics work that we did, we had a lot of patients who have blocked bile duct also, and we call them the BBP, which is the benign biliary pathologies. So they are non-cancerous, but they have different benign pathologies. And so we had that group as a control. So it will help us differentiate and say, if we identify in a group of proteins that are dysregulated, we want to make sure that these are only dysregulated in pancreatic cancer patients. So again, for example, if you look at this Venn diagram here, where we did pancreatic cancer versus benign biliary and pancreatic cancer versus healthy control, what we were trying to find is actually that group of proteins that we could confidently say that these proteins are significantly upregulated or downregulated in our pancreatic cancer compared to control this patient. So I think we always keep that in mind when we are choosing our patient population and our controls, that we identify patients who also have things like the blocking of the bile ducts, so that we identify proteins, metabolites, or even microbiome communities that are only key and unique to pancreatic cancer patients. I hope that answers the question.
Yes, sir. Thank you, sir.
Thank you.
So, any further questions from the side of the audience?
No, sir.
Nothing. Okay. So, Professor Neveke, it was a very wonderful lecture. If you see, I have a book with me. The book is written by Randy Posh. The book name is The Last Lecture. I was reading this book actually. This book is written by Professor Randy Posh, who was a very promising computer science professor at the Carnegie Mellon University, and he died of pancreatic cancer. Okay, he died. It was when he was diagnosed with the disease. It was a bit late, and he said he tried to do some of the treatments, but it was so aggressive that he passed within 3 to 4 months once the diagnosis was made. So it's a very timely discussion about pancreatic cancer and how it aggressive goes on, and especially the precision drugs, and also about the genomics of how this disease become aggressive. Thank you very much. Because of the conference team, I would like to give the warmest thanks for your time, for spending and giving us with the insights. Thank you, Professor Nagaki.
Thank you very much, Dr. Thomas. Thank you.
Thank you, Professor, for your insightful presentation. Now it is my pleasure to invite our second speaker, Dr. Rajesh Raju, Professor and Director, Center for Integrative Omics Data Science, Yanapoya Research Center, to deliver the second invited lecture. I request Dr. Ranjit Kumar to introduce the speaker.
Dear, so Professor is online. Professor Rajesh Raju is online, right?
Hello, am I audible?
Yes, yes, sir. First of all, let me give a brief introduction to you. It's my great pleasure to introduce the second speaker of the day, Dr. Rajesh Raju, who is a professor and director of the Center for Integrative Omics Data Science, the Yanapoya Research Center, Yanapoya deemed to be University at Mangalore. Dr. Rajesh Raju obtained his PhD in biotechnology from the Institute of Bioinformatics, Bangalore, in collaboration with Colombo University, Shimoga. He is a distinguished researcher in molecular biology and bioinformatics. Important contributions to biological data sources, signaling pathway analysis, and integrative omics. Dr. Raju has played a significant role in the development and analysis of signaling pathways in NetPath, a valuable resource for researchers working on cellular signaling networks. His contributions also extend to the draft map of human proteome, a landmark scientific achievement that was featured on the cover page of the prestigious journal Nature in May 2014. Throughout his career, Dr. Raju has been involved in the curation, review, and coordination of major global biological data resources, including the Human Protein Reference Database, Plasma Proteome Database, Pancreatic Cancer Database, and Triple-Negative Breast Cancer Database. His significant scientific work also includes the analysis of sequence variations in primary immunodeficiency disorders and the development of approaches for predicting novel disease-associated genes through international collaborations. He received an Independent Young Investigator Award grant from the Science and Engineering Research Board under the Department of Science and Technology, Government of India, and also represented India in the Indo-Japan Youth Exchange Program in Science. Currently, he is at the forefront of establishing an integrated data science unit focused on analyzing biological context dependent signature across multiple molecular layers, including genome, transcriptome, metabolome, proteome, and post-translational modification profiles. What makes Dr. Raj's work especially relevant to this conference is his integrative vision. The research shows how high-dimensional biological data can be transformed into mechanistic insight, disease understanding, and translational possibilities. I now request Dr. Rajesh Raju to deliver his invited lecture. The stage is to you, Dr. Raju.
Okay, so is the screen visible? Hello?
Not yet.
Is my Yes, what to do?
Yes, sir. It's visible now.
Oh, it's visible? Yeah.
Yes.
Okay. So I hope the screen is visible now.
Yes, sir.
Yes, yes.
Yes, yeah. So good afternoon, everyone. So I take great pleasure uh, to introduce one of the major topics that we have been working recently. That means towards illustration of post-translational modification specific regulation and function of the human proteome. And I thank the organizers of Biospectrum 2K26 for inviting me as a speaker for this international conference on recent advances in biological sciences, basically bridging research to breakthroughs. So what is a multi-PT? That means multiple post-translational modifications. The figure that you see is for a particular protein called fumarase, which is a metabolic enzyme that converts fumarate to malate. And currently, with current information available with multi-PTM data sets, let's say high throughput as well as targeted, including site-specific antibodies. So this is what is available today. And you could see major post-translation modifications currently reported on this and is mapped across this protein. Although we talk about at the protein level, this is the complexity that it adds to each of these post-translation modifications into these particular proteins. And of course, being phosphorylation, the major studied post-translation modification, and that is derived across various studies in different experimental conditions and contexts, and that dominated at this point. But nevertheless, you also have myelination, methylation, acetylation, succinylation, ubiquination, and many of them also accounting at specific regions. So which indicates that our information on the regulation of proteins or their functions are far from what we actually think, because each of them can give a context specific regulation or the even the stability, even the location, even the functions, along with other proteins, considering them as catalysts for interactions or any of other metabolic enzyme activities. So one of the major themes that I'm going to introduce is an overview of what are the different post-translational modifications associated with regulation of metabolic pathways. And as you know, metabolic enzymes regulate cellular homeostasis through coordinated anabolic and catabolic reactions. And post-translational modifications, of course, we believe act as molecular switches, controlling the enzyme activity, stability, localization, and interactions. The lack of a unified framework integrating multiple PTM landscapes across metabolic pathways limits our current understanding of their regulatory roles. Consider we derive phosphoproteome data sets, so you
Sorry for the inconvenience. I think there is some net issue. Please wait.
Hello.
Yes, sir. Sir,
Hello.
yes, sir.
Am I
Sir, I think there is a network issue. Hello, sir. Hello, sir.
Yes. Can you hear me?
Yes, sir, I can hear you.
Are my slides visible?
Sir, you're presenting the screen as such. Slides are not visible yet. Hello, sir. So the presentation is visible.
Yeah, okay. Okay, shall I go ahead with this? Okay.
Yes, sir. Yes, sir.
Yes, okay. So, as I mentioned, I hope all have reached to this stage where this clearly indicates that phosphorylation is one of the major post-translational modifications that are currently reported in metabolic enzymes. And not only them, you have methylation, ubiquitination, acetylation. Although they have been explored less, they also significantly contribute to major metabolic enzyme functions. So now, if you look into the top distinct post-translational modification across major 25 metabolic enzymes, you still see that many of them, like nitrosylation, they also pop up very significant. So which indicates that many of their functions are currently not known and needs to be explored. So relative to a particular post-translational modification, the assumptions and experimental evidence known is very limited at this point. And we analyze these PTM hotspot regions, basically the regions which spans within -7 and +7 amino acids of each of these post-translational modifications to see if within those regions other modifications also come across. And these hotspot regions we defined for major post-translational modifications in metabolic enzymes. And we also identified many crosstalk residues across these metabolic enzymes. So just to conclude the basic aspect, we had around 29 distinct PTMs currently associated with metabolic enzymes with respect to the current knowledge. And we have around 340 enzymes for which multi-PTM hotspots have been identified, and around 530 enzymes had PTM crosstalk regions, residues reported in them. And of course, phosphorylation predominates. Yet, we also did density frequency analysis, and we identified that what are the different enzymes which are regulated by multiple PTMs. And out of all these analysis, we identified 130 metabolic pathways predominantly regulated by post-translational modifications. Then we focused on the rate limiting steps. So currently for around 112 different rate limiting steps are reported, and then we identified major post-translational modifications for each of these metabolic pathways, like glycolysis or citric acid cycle, wherein the distinct post-translational modifications that have the potential to regulate these. So which indicate there is a crosstalk with other upstream enzymes which mediate these stealases or kinases or ubiquitinases that can come into play with relation to specific sensing or signaling mechanisms. So these are the key findings which I have already put up to. Now, coming back to phosphorylations or other major post-translational modifications. So we know that human kinome currently characterized with over 500 different kinases can form Am I audible now?
Yes, sir. Yes, sir.
Yes, sir, you're audible.
Oh, okay.
So it's better if you turn off your face cam and then present the screen only.
Yeah.
The net issues will resolve.
Yeah, I'll try. Visible?
Yes, sir, it's visible.
I hope it's visible.
Yes, sir.
Yeah, so I'll just press. So we have a landscape, the phosphorylation sites in the human kinome based on the high throughput evidence as well as low throughput, and those supported by multiple evidences. And this shows an overview of the current of the landscape of evidence-based human kinome phosphosites. And further, we have gone to the conservation across different families and seen the phosphosites and the, one of the basic questions that are being with the community for a long time. And this has led to the classification of these phosphosites in the kinome tree. You can visualize that many of the kinases now within the families, now we could distinguish based on their phosphorylation sites. And we believe that many of them actually contributes uniquely to their substrate specificity. And this is basically another way of representing it. And I hope the slide is visible. Okay, and then based on this, we have actually characterized this into a resource wherein now we can put in any of these sequence and then you can characterize these kinases or any specific fossil sites or mutation according to their position. And then when we look into multiple kinases, another major aspect that's just come into is the isoforms. Many of the isoforms that you see and this has led to analysis of intrinsically disordered regions in the human kinome. So, in fact, we analyzed this study with respect to 523 human protein kinases, and based on LSTM deep learning model, we came up with a new prediction of all these idea regions, and then this has led to identification of 2,863 disordered regions across 518 kinases, which were conserved, and their analysis was performed with respect to DFG, HRD, APU, all these motifs, and then this has led to the first or currently the better prediction model for ideas in the human kinome and could be extended to any other proteins. And the impact of many of these idea regions have been, few of them have been studied, but it is well known that most of these post-translational modifications, ideas forms many of these hotspots. And based on this analysis, we have also looked into the structural integrity and mutational impact, and then we have developed a resource called kin disorder, wherein you can put in any of these sequence and it predicts the potentiality of idea regions. And now coming into the biological translational impact, we have identified many of these fossil sites within these idea regions. For example, one of them that you have seen So, for example, one of the sites that you can see here is 540 S421, which is one of the major phosphorylation site reported in the. But there is no function known about, and it is outside the kinase domain. And this has been identified in so many as around 70 different phosphoproteome datasets, and also differentially regulated across major experimental conditions. So we do not have the facility currently to identify the functions of each of them based on site-directed mutagenesis. And even if we do so, so they are mostly in very different context-specific experiments. So we assembled different phosphoprotein data sets from distinct experimental contexts, and we identified their correlated phosphocytes in other proteins that frequently correlate with them, either uniquely in the sense when it is upregulated, other phosphocyte in another protein also upregulated. So likewise, we have characterized the positively as well as negatively correlated proteins in each of these phosphocytes, and then we looked for the cumulative distribution and their across data sets for all the statistical approaches to see the significance of their correlation. And the right side, you see distinct experimental conditions in which they have been reported or reported to be differentially regulated, including growth factor stimulations, inhibitors, and various other contexts. And finally, we happen to identify the site-specific impact on various functions. And together, it proposed that tau-1, the major phosphocytes, 421, 965, 445, are the major phosphocytes that are differentially regulated in them. And our analysis suggested that serin-9 is an autophosphorylation site of tau-1, which actually induces its kinase activity. And most importantly, 421 as one of the major sites associated genome integrity. And after publication of this paper, many other experiments now with the site-directed approaches have shown the similar results, which indicates that our approach towards defining these phosphocyte-specific functions are really valid and can contribute to many hypothesis-driven approaches in the future. Similarly, we have many other phosphocytes identified in many other proteins, and most of them among these three sites, based on site-directed approaches now recently, the researchers have identified that many of the predictions happen to be true. And then we also plan to develop many of these networks based on this approach so that any phosphoproteome, any other master modification, high throughput data sets could be analyzed. So basically, at Center for Integrative Omics Data Science, we look into global data integration and multi-omics for biomedical innovation. And then we develop signal transfer pathways. Those things I have not touched upon at this point because of lack of time. And this is our team. And so thank you all. And I apologize for the internet connectivity because something has happened here at this point. So I hope I was audible and I could take you through all the slides. So thank you very much.
Thank you, Professor Raju, for your wonderful talk, even though it was slightly disturbed in between, but it was very insightful. So audience, if you have any doubts, you can ask the doubts now to Professor Raju.
No, sir.
So any clarifications needed from the professor, from the part of the audiences, please ask.
No. No, sir, there's no questions.
Okay, please ask.
Sir, there is no question, sir.
Okay, fine. So thank you very much, dear sir, for your wonderful lecture on how we can use bioinformatics tools for the study of especially the difficult diseases. Thank you very much for insightful and scientifically rich presentation. The lecture was clearly demonstrated how integrative omics and biological data science are redefining the way that we understand the biological complexity and cellular pathways, disease mechanisms, and translational applications. Thank you very much, dear Dr. Raju. So let me give my concluding remarks of this session. The session has beautifully reflected the scientific depth and interdisciplinary ambition of the Biospectrum 2026. Dr. Rajesh Raju's second lecture highlighted the power of integrated omics, bioinformatics, and biological data science in mapping complex molecular systems. Professor Navak's lecture extended the discussion into a domain of translational cancer biology, biomarker discovery, and precision medicine, particularly in the context of population-relevant healthcare challenges. Together, these two lectures remind us that the future of biological sciences will be shaped by integration of experimental biology with computation. integration of mixed layers with clinical interpretation, integration of molecular mechanisms with therapeutic decisions, and integration of global scientific knowledge with regionally meaningful health solutions. And on behalf of the organizers and all the participants, I sincerely thank both speakers for enriching this conference with the scholarship, vision, and scientific leadership. I also congratulate the Department of Biosciences and IQC of the Macfast College, Tirula, for curating such a meaningful and academically vibrant platform. Thank you very much and congratulations.
I would like to extend my heartfelt thanks to Dr. Rajesh Raju and Professor Ekne Emmanuel for their inspirative sections. I will also like to extend my heartfelt thanks to Dr. Ranjit Thomas, FRC, who chaired the session. Thank you, sir.
Thank you.
With this, we conclude the lecture sessions. We shall now break for tea and come back at 3:30 p.m. for technical sessions.